So my name is John Adams. I'm Deputy Director here at Biosphere 2, and we're standing inside Biosphere 2's desert. Just like the desert, and you can see how large the space is, Biosphere is a remarkable research facility because of its scale. There's no other place in the world that you can do the types of earth and environmental science projects that the University of Arizona does here. We're studying how does water move from the mountaintops into the aquifer and what impacts that quality, how are coral reefs in the oceans going to respond, and how are our rainforests going to respond to temperature and drought. Hello, my name is John Adams, and I'm Deputy Director here at Biosphere 2. We're inside Biosphere 2's coastal fog desert. This type of a desert is different from that which we find here in the Tucson area. And so if you were to travel from southern Arizona down the Baja coast, So my name is John Adams. I'm Deputy Director here at Biosphere 2, and we're standing inside Biosphere 2's desert. Just like the desert, and you can see how large the space is, Biosphere is a remarkable research facility because of its scale. There's no other place in the world that you can do the types of earth and environmental science projects that the University of Arizona does here. We're studying how does water move from the mountaintops into the aquifer and what impacts that quality, how are coral reefs in the oceans going to respond, and how are our rainforests going to respond to temperature and drought. Hello, my name is John Adams, and I'm Deputy Director here at Biosphere 2. We're inside Biosphere 2's coastal fog desert. This type of a desert is different from that which we find here in the Tucson area. And so if you were to travel from southern Arizona down the Baja coast, you would encounter many of the types of plants that were incorporated into this biome. The unique thing is that you travel through several different desert regions in and of itself. And in this system, we've packed together plants that would typically not be found assembled together. For example, we have Joshua trees, we have cordones, we have bujums, and again, some of these specimens would never be found in such close proximity to one another, but because you're able to recreate ecosystems inside Biosphere 2, the designers were able to achieve this. My name is John Adams. I'm Deputy Director here at Biosphere 2. One of the really unique and institutional experiments that we have going on at Biosphere 2 under the management and ownership of the University of Arizona is LEO, the Landscape Evolution Observatory. We've constructed and completely repurposed the former agricultural space to now provide us an opportunity to better understand it rains in the mountains, how much water ends up downstream for you and I to use, and what impacts that quality. And we've had some really interesting finding because hydrologists will tell you that most of their models were set up and designed and the algorithms that are used in the 19th and 20th century. We have very little ways to test the validity of those. Well, LEO offers them a new tool for them to better understand those dynamics and hopefully be better predictors as what's happening to the hydrologic cycle outside. My name is John Adams. I'm Deputy Director here in Biosphere 2, and one of the really unique features of Biosphere 2 is our equatorial rainforest. Now, although it's not a perfect representation of that which you'd find down around Brazil, it is an assemblage of tropical plants inside of a completely controlled environment. What makes this unique is we've got plants that are only a couple of feet tall to plants that are upwards of 80 or 90 feet in height and have been maturing in this facility for nearly 25 years. This allows our researchers to make changes to look at how this entire system responds. And when we see a response, we have a much higher level of confidence that the response we see is attributed to the change and not that the fact that the system is going through some level of stabilization. Some of the research we're doing is it's predicted to get warmer and drier in the tropics. Well, how do you turn up the temperature or turn off the rain in an actual rainforest? You can't. Inside Biosphere, we can do that, and we can get a much better idea of the adaptive strategies these plants have in dealing with these extreme conditions. Hello, my name is John Adams, and I'm Deputy Director here at Biosphere 2. One of the truly unique features of Biosphere 2 is we have a million-gallon ocean. It was originally designed after a Caribbean reef environment, and early on, some of the most significant research came out of this area. And we were able to show a direct connection between increasing atmospheric CO2 and decreasing coral calcification. And what I mean by that is that the ability of corals to grow was significantly impacted in a negative way as CO2 in the atmosphere goes up. Today, we have a really unique opportunity as we have a group of researchers from around the world who are interested in repurposing our ocean to continue not only a better understanding of increasing CO2 and its impact on corals, but also how is that further compounded by the warming conditions that we see around reefs.
So we're going to talk about what is a coral. Now, corals are an animal that actually live in this calcium carbonate shell. They live in these small little pores of this shell here. And they have this really unique symbiotic relationship with something called a zooxanthellae, which is a small plant cell that lives inside of the tissues of the coral. Now, that's important because corals actually rely on those zooxanthellae for their energy. That photosynthesizing process provides them with the energy that they need to survive. So coral is an animal, lives in marine systems, and is most commonly found in coral reef environments. Corals are incredibly important for a couple different reasons. They're a place that 25% of the marine biodiversity that lives in our ocean, they depend on coral reefs, whether it's for a place to live, a place to be sheltered, or a place to find food. So that's a large amount of animals that depend on corals. Now, we as humans also depend on corals because fish depend on corals. We depend on those reefs being stable in order to continue to have food. The other reason corals are important is they're a huge part of economic development for a lot of countries. Countries rely on ecotourism. They rely on coral reefs, bringing people out to their nations and to their different places that are coastal environments. People go on vacation to coral reefs because that's what they want to see. So a lot of people rely on coral reefs for ecotourism. So inside of our Biosphere 2 ocean, we have some really cool invertebrates that are living in here right now. This that I have right here is a black long spine urchin. These guys are known for being voracious herbivores. They love algae, so they eat it at a very rapid rate. So we're just now in a trial period with these guys to see how they do, and they're doing really well. One of the things that we're trying to do in this ocean right now is decrease our levels of algae. So we have about 15,000 hermit crabs. So we have about 25 of these black long spine urchins. We also have about 20 strawberry crabs, and all those animals help us clean up this algae. They eat it really quickly. So we're doing that right now to try to decrease this algae in preparation for this coral project that we'll be starting. So this black long spine urchin is a really important part of our Biosphere 2 ecosystem out here. One of our other invertebrates we have inside of our Biosphere 2 ocean, along with the hermit crabs and the urchins, are these really cool little strawberry crabs. So this strawberry crab also loves algae. Now these guys have a really cool adaptation. So you can tell that they're bright red. Something that they have in order to kind of deal with that is they have these small little hair follicles you can barely see onto the top of its shell. Now those little follicles actually help collect algae. So you can see that it's got some green on its top. So that's a huge part of its camouflage method. They don't have to worry about as much worry about how red they are so they don't get preyed upon. Now that's not really an issue in our Biosphere 2 ocean. We don't have anything that's going to eat these guys. They've got a pretty great life. All they have to do is go around and they can eat all the algae they want and get to just graze in our Biosphere 2 ocean. So coral reefs, as cool of an animal as they are, react really strongly to different stressors. And we've known that about coral for quite some time. We started to see, as carbon dioxide increased in our atmosphere, we started to see coral having an invert reaction to that. They started to actually, their shell started to disintegrate because the ocean was becoming more and more acidic. The other stressor that's coming in now is that our oceans are getting warmer. They're getting warmer by 2 degrees Celsius in a lot of places. A lot of corals don't do well with that increase. Any kind of change in their environment really adds a lot of stress to their plate, and they're not able to grow or survive. They also have reactions where they actually expel their zooxanthellae, that symbiotic plant cell that lives in their tissue. And if they don't have that, then they're having to find their own energy. So these corals being under stress is a big problem. We've lost 30% of our Great Barrier Reef in 2016. So our coral health is going down, and we're losing corals at a rapid rate, to the point where we need to do something about it, and we need to do something about it quickly. So at Biosphere 2, we are working with coral researchers from around the world, from University of Hawaii, University of Miami, all different places, University of Australia, and we're looking at how we can prevent corals from leaving our oceans. So what they're doing is they're actually selectively breeding the corals that are surviving this increased temperature, the ones that are more resistant to these stressors. When they selectively breed those corals, they're making a supercoral, one that's even stronger than its parents. That supercoral is what is going to be surviving in our oceans. They're doing these tests in the laboratories now in Hawaii, and we're hoping this is going to be phase 2. We'll actually bring those supercorals out to the Biosphere 2 ocean and continue to do testing on them before we do anything in the natural systems. Out here at Biosphere 2, we are working with coral researchers from all over the world, from the University of Hawaii, University of Miami, even some universities in Australia. And we are collaborating with them to figure out what we can do to save the coral reefs. Now, they're doing some preliminary testing. They've started doing some testing in the University of Hawaii laboratories, where they're taking corals that are more resistant to this increased stress, the increased heat that we're seeing in our oceans. The ones that are surviving this increased heat, they're actually taking them and selectively breeding them, crossbreeding them together to create a supercoral. So they're essentially speeding up evolutionary processes to create this supercoral that will withstand the increased temperature. Now, we're going to be doing that in our Biosphere 2 ocean right behind me here. We'll be putting those corals inside of this ocean and being able to do different modifications to it, different testing to see how these corals are reacting. So they're doing it in phase 1 in the laboratories now with just these little coral fragments. We'll be having more of a large reef system put into this Biosphere 2 ocean to even elaborate on the research even more before we try it in the natural systems. everyone. Welcome to our Facebook Live. My name is Katie Morgan. I am Manager of Marine Systems, and today we're coming to you live from our Biosphere 2 mangroves. This system has been around since the original crew members were inside of Biosphere 2. These mangrove species, we have 3 species of mangroves. We have black mangroves, red mangroves, and white mangroves. These mangroves were brought in actually from the Everglades all the way over to Arizona in some trucks. That was quite an adventure as we understand it. They actually got stuck at the border of Arizona trying to get these plants in because Customs didn't have mangroves on the list of species that could be let into Arizona. So they kind of got stuck there for a little while until they got the correct permits and permissions to bring them over here to Biosphere 2. And then they were quarantined, these mangroves were quarantined until the space was ready for them to be put in. And the unique thing about this biome is that not only were these trees brought in, but so was the water and the soil and the microbiome that comes along with that soil and water. So it's one of the most full biomes that we have out here. It all came from one place right here in front of us here. And you can tell the difference between these trees. So right off over here are red mangroves. You can tell because they've got these more aerial roots. So these roots are higher off from that water's surface. And then over on the other side are our black mangroves. You can tell those roots are different. They look kind of like fingers emerging from the water there. So those are how you can identify the differences between those 2 species of trees. And like I said, we also have some white mangroves in here. Those white mangroves are on the other side of this marsh mangrove system. So you can't see them from the perspective that we're at, but they are. There's a couple trees that are on that side. Now, when these trees were brought in, none of these trees were taller than 5 feet. So you can tell there's been quite a bit of growth in the 30 years of this system being here. These trees have done really well in this system. Mangrove environments in general are incredibly resilient ecosystems. So the water that's surrounding us right now is what we call brackish water. So it's a combination of salt water and fresh water, and its salinity is quite a bit lower than the oceans, but it's kind of that middle ground between the fresh and the salt water. So these trees are actually living in saltwater right now, which is a very unique thing. Not a lot of trees or plants in general can do that. So they're able to adapt to this saltwater environment by actually pulling that water into their roots, and they extract the salt on the back of their leaves. So they don't use that salt for any purpose. They actually just go ahead and get it out of their system, and they're able to extract it on the back side of their leaves. Now, originally, when this mangrove system was put together, the idea is that we would have a freshwater marsh that would connect to the saltwater marsh that then would connect to our ocean system. Now, this makes a lot of sense when you think about it because the oceans are connected to mangroves and the natural systems around the world. But when you think about the scale of Biosphere 2, our mangrove system scaled to our ocean system scales are not different enough for that to work. But they didn't know that yet until they tried it. So they actually did connect these systems for a very short amount of time until they quickly realized that the nutrients in the mangroves was way too high to be putting into the ocean for it to be successful. So these systems are completely separate today. Our ocean and our mangroves do not have any interaction, but they did in the early 90s for a very short amount of time. Now, there's a lot of research that has come out of this ecosystem here. There's been research looking at the differences between a system in a closed environment, like we are inside of Biosphere 2, compared to those trees that are out in the natural systems in the Everglades. They've looked at different things, including leaf litter. So how does leaf litter decompose in this kind of system? You can see a lot of the leaf litter down below us here. So they looked at those decomposition rates in the leaves. But they've also looked at levels of insects and insects eating and destroying some of these plants. Now we have very little insect life in here that is negatively impacting these mangroves. So they've been able to do studies looking at how the mangroves in Florida are having stress reactions, and how that might affect the anatomy of the plant, versus our mangroves in here, which have very little insects that are destroying their system. We have a unique collaboration with Patrick Kangas from the University of Maryland, and he is actually, for many years, he's been involved with the project since day one. He helped actually design this system, but he's been bringing out students from the University of Maryland to work with us here, and has built an algae turf scrubber system. Now, algae turf scrubbers have been around for a long time. It's actually how we controlled the nutrients in the ocean initially in the first experiment. We've moved on to some more modern technology today, but algae turf systems are really beneficial for areas that have high nutrient loading. So mangroves are a really great example of a system that has really high nutrients. And in order to kind of extract some of those high levels of nutrients to increase productivity, they're able to use these algae turf scrubbers to grow the algae, and then you pull that algae off of it, kind of remove some of those excess nutrients to keep the system healthy. And then those algae that's being pulled is being looked at for things like biofuel and food sources. So there's a lot of innovative research going on looking at how to use mangrove systems and technology like these algae turf scrubbers to think about how we are better in the future. Now, a lot of people ask when we're in here, what kind of animals do you have in here? I think that's a common question we get throughout all of our biomes. Now, I mentioned that they brought in soil and water and the microbiome from Florida, and that is true. Along with those came we have two species of fish in here, a killifish and a mosquito fish. The main fish that we have in here today is our killifish. We don't see many of those mosquito fish in here anymore, but we do have those swimming throughout. But we don't have a lot of the other traditional animals that you might see in mangroves, like fiddler crabs or sponges, or any kind of like a lot of juvenile animals. So mangroves are around the world very beneficial for a lot of our juvenile marine animals. A lot of fish and invertebrates actually go and lay their eggs amongst these roots. You can see how protected these roots may be. If those animals are coming in and burrowing in, laying those eggs, it allows a protective area for these animals until they are big enough and strong enough to go back out into the natural systems. Now, mangroves also play a couple other really key roles around our world today. You can tell these root structures are all up in this soil, so they actually are really great at preventing erosion. So having these complex root structures keeps all that soil in place. And if you think about it, where do you find mangroves? Well, you find them along our shorelines. And so they're really important for when we get big storms, hurricanes. They keep our shores intact. Now, this has been an issue as we think about developing our coastlines. I mean, most people, when they go on vacation, they have an option to stay right on the beach or 2 miles away from the beach. They're most likely going to want to stay right on the beach, at least I would. So a lot of our mangrove systems have been destroyed because of that, because of development. And we've really been able to put in protective measures to make sure that no longer happens, because not only do these mangroves protect our land, keep it from erosion, and protect those baby marine animals, they also are really great filters for our ocean. So these roots take up tons of excess nutrients, whether it's from pollution or just excess things that are coming from the ocean, and filter all those things out so that the oceans are cleaner and our mangroves are cleaner. So there's so many important things that these mangroves do, and we're lucky to have a system like this inside of Biosphere 2 that we can continue to use for research and compare a system that's been under glass for almost 30 years versus our natural systems, which are looking quite a bit different today. So it's a really unique research opportunity for us today. So thank you so much for joining us. It was a pleasure to bring you into the mangroves today. I do want to give a shout out. We are going to be doing a virtual Earth Day event. Earth Day is coming up on April 22nd. So keep your eyes peeled. We're going to be posting some information about that early next week. So we hope that you're able to join us for that. And we'll see you live from Biosphere 2 next week.
Good afternoon, everyone. My name is John Adams. I'm Deputy Director here at Biosphere 2, and I'm welcoming you to our daily live session on our social media platforms. And today we've got a real treat for you. Biosphere 2 has two large geodesic domes that are called the lungs. And they're probably one of the most remarkable engineering features of Biosphere 2. Now remember, Biosphere 2 was originally designed as a completely sealed environment. No material exchange, and the only thing that would move in and out of Biosphere 2 under its original design would be energy. Now what happens when you have a large mass of air inside of a fixed volume and you heat it up? Well, what happens is that means the pressure goes up. And conversely, when you cool that air mass down inside, then the pressure goes down. Well, the engineers quickly determined that if they didn't account for these changes in pressure, you could compromise the integrity of Biosphere 2. It could explode, or it could implode. And so what we're going to do is we're going to walk you down into the lung. But first, what we're going to do is to get to the lung, we've got to travel through a tunnel. We're going to walk you down to this tunnel, and there are two of these. The one we're going to go in particularly goes underneath the road that connects the desert and the lung. And so why don't you go ahead and follow me down the tunnel, and then we'll go into the lung chamber itself. So this tunnel is pretty cool. It's got some great acoustics. When we did have school children here, this was probably one of their favorite places. They would really like to let out some noises because the reverberation is overwhelming. But, you know, the Biospherians, when they originally designed Biosphere 2, thought of everything. So, not only would air travel in and out of this tunnel, but we're also, as you can see, on a downward slope. And this slope starts in the rainforest and works its way all the way down to here. So, if you're inside Biosphere 2, and one of your water pipes breaks, you don't want that water just running any place. You want to be able to try to collect it. So everything would work its way down into the lung where we have a large reservoir to collect all that water as well. So it was really ingenious how this particular space has many different uses. So as we go through here, you got to watch your step in your head. So let me turn on some lights in here. You can actually get an idea of just how big this chamber is. So, I mean, I think the most common reaction we get from people in here is, wow, and how cool it is. I mean, it just looks like something out of this world. Now, so I know many of you have got lots of time on your hands. It's a good thing and a bad thing, but what we'd like to do is if you get a chance, go on to Google Earth or one of these aerial views of Biosphere 2, and if you don't know what I'm referring to when I say the lungs, there's two geodesic domes, one on the southern end of the structure and one on the western end of the structure. And that is what the lungs are, these geodesic domes, and we're standing inside of one of those. And so the way this works, it's really sort of hard to imagine. But again, remember, as air expands inside Biosphere 2, it's got to go somewhere. Because if it doesn't have an opportunity to be released or compensated for, you could create so much pressure that you could push the glass out. You could explode the biosphere. And so what the engineers and designers did is they knew they didn't want to allow air to move in and out of the facility because remember, it was materially closed. But what they did want to do is they wanted to compensate or account for that. And the way of doing that is they created essentially a counterweight or a large diaphragm. And that diaphragm functions inside the chamber we're standing in right now. And it would go up and down to increase and decrease Biosphere 2's over volume. And what do I mean by that? So what you see here is a large aluminum dish. Attached to that aluminum dish is a flexible rubber membrane. It's black in color. And it is fastened to a fixed wall, a wall that doesn't move. Now, what happens is that material, that black membrane, is called Hapalon. It's a geotech membrane. If any of you know one of these Zodiac boats that you oftentimes see people using to move back and forth in the ocean areas is where they're most commonly used, it's that same type of really heavy rubbery material. It's attached to that aluminum dish. The combined weight, now get ready for this, is 40,000 pounds, okay? And remember, there's two of these. So essentially, when both lungs were working inside Biosphere 2, we had 80,000 pounds sitting on the air mass inside Biosphere 2. And what's really incredible is on the backside, the area that we can't see, there's no crane, there's no hoist, there's no winch that's lifting this 40,000 pounds up and down. The only thing that is moving that 40,000 pounds, 20 tons, is differential air pressure. So again, as that air expands, it's got to go somewhere. This is a path of least resistance. It pushes this whole structure up. As it goes up in height, and it can go all the way up to 50 feet. So right now, we're probably somewhere in the neighborhood of about 20 feet up. So imagine, another 30 or so feet in height that this can go up. And then at night, as the air cools, and you don't have as much pressure in Biosphere proper, this pushes down and pushes air back in. So the facility was always maintained at a constant positive pressure so that anything, and there were leaks, as you can imagine, it wasn't completely airtight, although they achieved a remarkable leak rate. Air would be pushed out, and that was by design, so that this facility was positively pressurized. So just like an airplane or just like the International Space Station. Now, what is harder to see is there's a big reservoir here. And this was Biosphere 2's primary water storage location. So the water cycle would have started and ended here. So right now, this pool will hold about 200,000 gallons when it's filled to its capacity. And again, originally the water cycle would have started and ended here. Now what we're going to do is we've sort of redesigned or re-engineered how the lung works in the sense that we've tricked it into being up in the state. And we've used physics and we've used differential pressure to achieve that. And we're going to take you over to a door. We're going to open that door. It's going to breach the seal. And we're going to watch this start to slowly come down. So this is one of those airlock doors. I'm going to open it here. What we're going to do is we're going to walk across. And I'm going to stand here. So by standing here though, it's going to give you a relationship because you're going to see this thing coming down. In just a few minutes, I will actually be able to touch this leg. And I'm going to go ahead and hang on it. So I'm feeling really strong today, but I'm going to be able to move 40,000 pounds here in just a minute. Because again, remember, it's just all sitting up in the air as a result of differential air pressure. So for those of you that really like physics, think about, and the law that most commonly applies here is the ideal gas law. So you got pressure, volume, temperature, you've got a constant in there, and then you got the number of moles of air that we have, or the weight of that. So again, that's what you would be used to calculate. If we know what the volume is, we know what the change in temperature is, we can determine, for example, what the pressure change is. And again, as temperature goes up, pressure goes up because our volume is fixed. As temperature goes down, our pressure goes down. So you can see how quickly, before I couldn't even reach it. So now what I'm going to do is I'm going to pull down on this thing. And so we just moved 40,000 pounds. Now you may be wondering what these chains are. So under the original design criteria, as you could imagine, if this thing got too high, the lung got too high, got above its 50 feet maximum height, you wouldn't want it to continue to rise up and rip the membrane. So the chains pull down, they would have broken a piece of glass and it would have allowed the air to come in to equalize the pressure. So again, they had safeties put into place to ensure that they wouldn't compromise this particular structure, Biosphere 2. So now what we're going to do, let's come down quite quite a bit. We're going to walk out this door here, and I'm going to show you just how much force and air is being pushed out through this small air hatch. Now you may ask, well, how are we lifting up 40,000 pounds? The biosphere is no longer sealed as it originally was designed because of the research that we do. But what we are doing is we are creating a vacuum in this space here, pulling air out. That vacuum is enough to create enough differential pressure to lift up the lung. So again, join us tomorrow. We'll have another exciting feature for you from a unique location inside Biosphere 2. Again, my name is John Adams. I'm Deputy Director here at Biosphere 2. We wish you all well. We hope everyone is safe in these really uncertain times, and we look forward to seeing you again tomorrow.
All right, Aaron, you're live.
Hey guys, my name is Aaron Boucher, and right now I'm filling up what we call an Oya bottle. The concept of this bottle is about 4,000 years old. People have been using terracotta clay as an irrigation source for their gardens for thousands and thousands of years. And as I fill this Oya bottle up, you'll start to slowly see some black spots. Let's see if we can find some. There's one down here. Now this clay is going to start to soak up the water, and it soaks up the water through these micropores. And this is pretty cool because the clay eventually gets saturated, but then it does a really good job of hanging on to that water until there's a strong enough pull to extract the filled bottle, the water inside the filled bottle, to extract it outside of itself. So some of these forces that pull water out of this bottle, which we call an Oya, O-L-L-A, are soil tension. They call it the matrix potential, as well as plant roots, thirsty plant roots. So this creates a kind of unique irrigation source, and it's got a glazed top because that helps prevent evaporation from the bottle when on the exposed surface. Now, I do want to say that this technology was not invented by us. It's been used by native cultures originating from South America, Mexico, the Middle East, and the steppe regions of Asia for thousands of years. Now, these more modernized versions of the Oya have been developed by one of our partners, Cutting Edge Ceramics, and they've also developed some other styles. But we use them in an experimental garden here at Biosphere 2 as a way to teach students and K through 12 or undergraduates about how soil hydrology works, alternatives for small-scale agriculture and gardening, and also learning new ways to study soil and some of the forces at be with different soil types. So we actually designed 4 Oya garden beds that have different soil types in them. And as you scan through these different Oya beds, you'll notice that some of the plants are doing quite well and others are struggling a little bit. And we irrigate them with the same amount of water, but the other factors at play here are nutrient availability. And this bed here is Leo soil. We had a live session from Leo a couple weeks ago, and you guys will remember that that's volcanic basalt, crushed down to a tephra-like substance. Now, this basalt does contain some nutrients, but it's quite lacking down the stretch in terms of nutrients that are available for plants to use. Now, what we're studying on the Leo slopes is, okay, how does this soil break down through the addition of rain events and climate events, how does that break down to form more nutrients that would actually allow this garden to grow 2-3 times as big, to really thrive? But some of these species are stunted, and you can see some of their leaves are yellowing, which is a sign of missing nutrients. But as we move down the line here, we have other soil types that are either Leo cut with compost. We've got a bed that's just compost that from our partners Tanks Compost down in Tucson. This chart is struggling a little bit, but I think it's just starting to heat up. And what we do, as you can see, is we've got our Oya bottle buried in the ground. We fill it using a hose, and then I cap it with the top. And these plants have found this water source, and they found it probably 4 or 5 months ago when I planted for winter. And once they find that source, they're gonna latch onto it and wrap their roots like giving it a big hug. But their roots are also gonna keep scanning for water as well. So what we have going on in these beds are three-dimensional networks of root systems that have traveled quite a long way to find those water sources. And let's pick one where you can actually see some of the roots, maybe down here, Katie. We created these plexiglass visualizations to show visitors what it looks like on the subsurface. You can see roots, and because it is plexiglass and there's available sunlight, you see a little bit of growth here on the wall as well. This is a little bryophyte algae, but you can see these roots finding the Oya and then even scanning farther outward for water sources. And they're industrious, so they're gonna find water where they can. And in this basalt tephra, that soil is gonna hold on to water quite well. So we've been using these gardens to teach K through 12 education groups, undergraduates. We'll bring them out here with photosynthesis instruments like the Lycor 6400 or soil moisture probes to test the soil moisture of the soil as it moves away from the Oya, to see how far that water starts to expand out of the bottle. And if we come back to our bottle that I originally filled up, we can see that it's now saturated with water, and it feels cool on the outside. And that's something of note because before this was used as an irrigation technique, it was also used as a method for cooling homes. We get this really nice evaporative cooling effect using this terracotta clay, and it's not just for irrigation. So we've planted tomatoes, peppers, herbs like rosemary and mint, and some of our chard has done really well. Radishes have done very well. So there isn't much that you can't grow using Oyas, but there is a limit of scale, right? We can't irrigate a 20-acre farm with Oya bottles. The largest scale that I've seen this method implemented on was about a quarter-acre farm, and they were actually using terracotta pipes, like long pipe systems underground. But as you can imagine, if you have a leak in that system, you'd have a hard time finding where that leak is. So these small-scale systems with Oya bottles are just perfect for your backyard and small-scale agriculture.
Hey Aaron, we've got a question from Jen. She missed how does she get one of these bottles, or maybe you can tell her about how to make one herself.
Yeah, absolutely. So like I said earlier, we partner with a group called Cutting Edge Ceramics in Tucson, and you can go to their website, cuttingedgeceramics.com, to order Oyas. You can also, when we're open as a facility, buy some of our Oya bottles in the gift shop. But there is another method for creating these Oyas in your own kind of DIY. And why don't you follow me over here? Right here we have a DIY version of the Oya bottle. You take 2 6-inch pots, from Home Depot. I used a piece of tile and some Dural 2-part epoxy to shut the bottom hole. And then I used Gorilla Glue to glue these two pots together. Thank you. And then we've got our watering hole at the top here. And this works quite well. One of the projects that I did with students several summers ago with our Summer Science Academy, we looked at how these different types actually perform. So we have our Oya bottle, we have our large DIY version of the Oya. There are also things like Oya balls that are gravity-fed systems, and I don't know if I have one. Maybe I do. Yeah, this is a little cutaway of an Oya ball. And what people do with the Oya balls, this is a design by Cutting Edge Ceramics. They daisy chain about anywhere from 5 to 10 to 20 of these balls together in their garden. And a single plant will wrap their roots around one of these balls and kind of call it home. So we have several different types of Oyas. And what we're working on at the Biosphere with our student groups is to actually generate some data to say, how do these different types of Oyas perform over time? So as we continue to collect that information, we'll be sharing it with the public as we move forward. Any other questions, Katie?
It doesn't look like it.
So yeah, we'd like to thank Cutting Edge Ceramics. They actually donated some of these Oyas toward our education program, which was very generous of them. And tune in for more from our Oya garden as we start to move towards a summer planting. Ask any questions you have and I'll do my best to answer them in the link below. And hope you guys have a great day.
Hello and welcome to Biosphere 2 Live. My name is John Adams, and today I'm going to talk to you about our amazing rainforest that we have inside this remarkable facility. So it's kind of odd to think about an equatorial rainforest here in southern Arizona, but that's exactly what the original designers and creators of Biosphere 2 created and constructed here. The rainforest here is approximately 144 feet on a side and 91 feet tall at its tallest point. So that's about a ½ acre in size. What you can't see, and what's really remarkable, is complete with a stream, a waterfall. There's a central mountain out of sculpted concrete that resembles or looks like rock. In fact, this structure is very realistic. It has moss growing over it. And then you can see the area was actually heavily vegetated with a number of different plants inside the system. And so one of the things we often get asked is, well, how many plants are inside Biosphere 2's rainforest? And so originally there were over 400 species planted in this system. Now today, we just recently had a grad student from the University of Arizona who finished up surveying, and that number is just a little bit over 90 species. Now everybody sort of takes a step back and goes, that's a huge loss. You know, what is happening with the system? Well, that was exactly what was anticipated. And in fact, the reason that they packed in so many different species in this rainforest was specifically to see what would happen, to see actually which species were going to survive, but also the hope would be is at the end of this self-organization period, we would still have a very diverse community, which is what we feel we have today. Now, one of the other questions we get asked, and this gives me a good moment to mention that if you do have questions that I'm not answering or covering and you've always wanted to ask, please go ahead and put them forward. I've got my colleague Katie Morgan and Jason who are monitoring the feed. They'll go ahead and relay those questions to me, and I'm happy to answer them as we go through. If by chance we don't get an opportunity to answer your question, we will be responding to comments, and we will also provide any critical links that we think will help you further understand biosphere. So, the species that were put in here were selected primarily from the New World tropics, so that area around the Amazon basin there in South America. Additionally, what they did put in is they packed in as many different types of insects that they could get, not only those that were found sort of regionally, but also those that were found locally here. There were a few reptiles and amphibians, and today there are still remnants of those. Every now and then, occasionally, we will see a tree frog in here. We might see a lizard, and it's called an alligator lizard. There are spiders if you look around. Now, if you dig in the soil, you're going to find a really diverse community of organisms. You'll find roundworms and flatworms. You'll find some millipedes. If you come in here at night, and oftentimes this gives people sort of the creepy crawlies or the chills, because this entire place is run with and run by roaches. And so we have a number of different roaches. And you go, well, why did they put roaches inside? Well, they put roaches inside because they are critical in helping to break down material and biomass. One of the things for this rainforest that we don't do is we don't come in and fertilize it. So we are dependent upon the system naturally recycling the nutrients back into the soil for the plants to take up, like those that I'm standing here in front of. Now, when you think about Biosphere 2 and its original history, we hear so much about some of the challenges that our original group faced. And one of those big challenges was decreasing oxygen, increasing carbon dioxide. Well, the rainforest here inside Biosphere 2 was one of the big culprits of this. And what I mean by that is the soils that I'm standing on and that these plants are growing in, they did not have the opportunity to go down to the Amazon basin and collect them. Soil scientists said, well, if you can't go there, maybe you go to Georgia and bring over, and trucks, or actually come by train, bring those soils here. They couldn't do that. So what they had to do, they had to use local amendments. They had to blend them with the best of their knowledge, using a lot of the scientific data and scientific experts in these areas to create this rainforest soil. They did a great job. They created a very rich material, and that material was designed to support this intensive growth of plants that they envisioned was going to take place and happen. But what it also did is it provided a great environment for microbes. It was extremely rich, and therefore these microbes flourished. And in fact, what ended up happening is the microbes would take in oxygen, they would give off CO2. And that pace was much faster than these plants could in turn take in carbon dioxide and conversely split water and give off oxygen, so that there would be a balance. And so the soils, in particular in the rainforest and also the agricultural areas of biosphere, contributed heavily to this atmospheric imbalance that was experienced. Again, for those of you who are just joining us, my name is John Adams. I'm Deputy Director here at Biosphere 2, and today we are coming to you live from inside Biosphere 2's rainforest. Now, we've done some remarkable research inside Biosphere 2 rainforest, and I hope that you will join us here in the coming weeks, because we are going to hear from our research team as well as the managers who run this system inside. Now, when you look around Biosphere 2's rainforest, again, one of the things that people notice is there's not a lot of sort of animal life. So you're not going to see a jaguar coming through the lower floor. You're not going to see monkeys traversing the canopy inside this system. Those types of large vertebrates would have been very difficult, if not impossible, for that original group to put inside here. What they wanted to do is create a system that would be self-sustaining and would not necessarily need a lot of help from those who were originally managing the system, and therefore why we don't have large mammals or even other large vertebrates in the system. Now, most of the plants that I'm standing in and among, they came in at about 10 to 12 feet, maybe 15 feet in height. We have now plants that are touching the very top reaches of biosphere. So they're in excess or close to 90 feet tall. We've got them spaced throughout, but we have had a very dramatic loss in those species. And one of the things that's very interesting is that this is probably the hottest rainforest that you're ever going to be in. And that's allowed our researchers to do some interesting research, but also is why we believe that some of the species that were originally put in here, so some of these orchids and bromeliads that require very narrow temperature and very strict humidity ranges, did not survive. But the plants that we do see surviving, they emit certain compounds that give them some adaptive characteristics and traits to allow them to deal with warmer temperatures. And so our research team in part is studying, and you're going to studying these traits, and you're going to hear more from them in the future. So this about wraps up the presentation today, but what I want to do is, again, if there's any questions out there, this will be a great opportunity to take them. Otherwise, we look forward to seeing you in the coming weeks. Next week, please be on the lookout for our Instagram and our Facebook and our Twitter posts. We will let you know when our rainforest manager, Jason, will presenting from inside here. And what he's going to do is describe what it actually takes to manage and run a rainforest inside of the closed environment of Biosphere 2, and really talk about some of the unique traits of the plants that we have inside. If you're interested in learning more about Biosphere 2, we have a great open course, and it's called a MOOC, a massive online course that you're more than welcome to take. We will provide a link for you to click on. And then if you'd really like to get an in-depth understanding of Biosphere 2's rainforest, I encourage all of you to participate in our researchers tour. So the 20th of this month, which is a Saturday at 2 p.m., we offer a rainforest researchers tour. It's going to be led by Dr. Joost van Haren, and he is going to talk to you about all the different aspects of Biosphere 2's rainforest, what it took to create it, what we have today, and how we're going to be using it from a research perspective to better understand the changes that we're seeing in the rainforest around the world. And that will also, we will also provide a link on our website for you to view. So again, thank you very much. Again, my name is John Adams, and thank you for participating in Biosphere Live.
Welcome to LEO, the Landscape Evolution Observatory. That's a big name for a big experiment. In fact, LEO is the biggest earth science experiment ever built, and it's all taking place at the University of Arizona's Biosphere 2, a world-renowned facility for earth science research. Earth science is the study of how our Earth systems work and interact, systems like the water cycle, and the carbon cycle, and the energy cycle. There are so many parts to earth science, and so many scientists who study the different parts, but all of them, from scientists who study geology to water to ecosystems, have realized that each of these systems is deeply entwined with the others. To understand how our natural world works, we need to study how all the systems and cycles dynamically interact.
Hello and welcome to Biosphere 2's virtual EarthFest in commemoration of the 50th anniversary of Earth Day. My name is John Adams, and I'm Deputy Director here at the University of Arizona's Biosphere 2. We have a great lineup for you today, and we're hoping that you'll be able to join us for all or part of it. We're going to kick the day off with a 5K virtual run around the historic Biosphere 2 property. If you've not seen the property, there are just some spectacular views, not only of the landscape, but perspectives of Biosphere 2. We're then going to give you a virtual tour of Biosphere 2 through our visitor center movie. And then we've got a great set of panel discussions that we hope you will join us for. We've got two of our lead researchers in our rainforest system who are going to talk about an experiment they just completed where they gave the rainforest an excess of a 70-day drought. We're then going to hear from two former biospherians who were not only instrumental in building Biosphere 2, but actually lived inside for 2 years. How relevant is that today? And finally, we're going to hear from two members of our ocean team who are going to tell us about the exciting renovation projects they are doing in our ocean system and the relevancy that the research that we're going to be doing in this system is going to have to help shed some light on this coral crisis we're seeing around the world. And you think of an ocean in the desert, you think, we don't have an ocean in southern Arizona, but we do right here inside Biosphere 2, and it's 1 million gallons. So we really hope that you'll join us for those panel discussions. And then finally, we're gonna wrap it up and we're gonna hear from a group of K-12 school children who recently competed in SARSEF's Science Fair. And what they're going to do is they're gonna tell us a little bit about the winning projects and why they selected that particular topic. Thank you for spending a portion or all of your day with us on this virtual EarthFest at the University of Arizona's Biosphere 2. Happy Earth Day, and we hope you all stay safe. Good morning, everyone. My name is John Adams, and I'm Deputy Director here at Biosphere 2. I'm really excited this morning to show you and take you on this virtual run of the historic Biosphere 2 property. Now, although you can't be with us here today, we really hope that you'll be able to join us in the fall or early spring when we will be able to reschedule this. So stay tuned to Run Tucson's website, and they will provide all the necessary information. But why don't you go ahead and join me now, and we'll go through. This is a great place to just sort of take a stop because you're running by the historic B2 Ranch. We call this the Ranch House. It dates back to the 1920s, the actual original homestead by a dentist in Tucson by the name of Dr. Lackner. And so he had a small cattle ranch and used this as a hunting lodge at that time. So this course offers just some spectacular scenery. Over here to the right, you have the Canadalo drainage with the Catalinas rising up. I think some of the most spectacular scenery in all of Tucson. And then all you have to do is look over to your left and you've got Biosphere 2, and this is really a unique angle that most people don't see. Well, welcome to the historic Suffolk House on the Biosphere 2 property. This is another
How does life form, develop, and reproduce? What is the origin of our universe? What is the future of our planet? The curiosity behind such questions is with all of us from an early age. Through science, this curiosity has led to experiments created on the grandest scales, from building giant telescopes in space and on mountaintops, to launching a spacecraft that will bring back samples of an asteroid. One of these grand experiments is here in southern Arizona. Owned and operated by the University of Arizona, Biosphere 2 is big science. In fact, it's the world's largest earth science experiment. Can you imagine a laboratory that holds a rainforest and an ocean? That is what Biosphere 2 does. It contains biomes that simulate Earth systems on a massive scale, providing a home for the huge questions asked by chemists, biologists, ecologists, and other researchers who work here to understand our planet. The physical size of Biosphere 2 is matched by the ambition of its educational mission, to show how science, technology, engineering, and mathematics are vital to our future, and to inspire new generations of scientists to improve our lives through their insights and innovations. Welcome to Biosphere 2. Biosphere 2 is a sophisticated model of our planet Earth. It's easy to take the Earth for granted. We are born here and live here all our lives. Sometimes we might look up at the moon or stars and wonder about other worlds, but there is plenty to marvel at right here on our home planet. The Earth is 4.6 billion years old, but it is constantly changing. We can see these transformations in the solid part of the Earth, the geosphere. From dramatic forces, such as volcanic eruptions and earthquakes, to erosion slowly wearing down a mountain, the geosphere is constantly being reshaped. We can see these changes in the fluctuating levels of Earth's water systems, its oceans and rivers and underground reservoirs, its hydrosphere. When we look up predictions for how hot or cold it will be, whether it will rain or snow, we are seeing changes brought about by the cycles of clouds and air currents that make our weather in the atmosphere. And when we look at the enormous variety of forms within the biosphere, the arena of life, we witness the result of thousands of generations of genetic adaptations to a changing environment. This is the inspiration for the name Biosphere 2. Earth science is the study of how the geosphere, the hydrosphere, the atmosphere, and the biosphere all interact in a complex system. Biosphere 2 is a living laboratory where scientists examine these interactions in a controlled environment. You can't put the entire Amazon rainforest under glass and control it. But the rainforest at Biosphere 2 is contained, so you can manipulate the water and temperature conditions to conduct experiments on the entire ecosystem. Then you can observe and measure the impact of changes, say simulating a drought, everywhere in the system from an individual leaf on a tree to the health of the whole forest. There are 5 very different biomes under glass at Biosphere 2, including the tropical rainforest. The ocean biome contains 700,000 gallons of water. The mangrove biome is composed of 2 major types of wetlands, a grassy marsh and a swamp filled with mangrove trees. The desert biome simulates an arid scrub ecosystem in a coastal climate, sometimes called a fog desert. The savanna biome, with a stream and waterfall, is a transition zone between the desert and the rainforest and is planted with many grasses and edible species of trees. Beneath these biomes is what's called the technosphere, more than 3 acres of electrical, plumbing, and mechanical systems that regulate the temperature, airflow, and humidity levels. The Energy Center complex is the hub for all control, electrical, and mechanical systems necessary to sustain Biosphere 2. Researchers use the unique resources of Biosphere 2 to design experiments that address real-world challenges, problems that can affect the quality of life across our planet.
We have an exciting experiment ongoing here in the Biosphere 2 tropical rainforest. An international and interdisciplinary team of scientists is descending upon the rainforest to conduct a large-scale drought experiment. And we're doing this by using this unique facility where we can simply turn off the rain. After we do that, we're going to track how and where microbes and plants are continuing to get their water during drought. We're going to look at how carbon continues to cycle in a forest ecosystem under drought. And finally, we'll be tracking the different types of compounds that microbes and plants produce when they're under this type of stress. And this is going to help us better understand the future of our tropical rainforests around the globe. So in this controlled facility, we can decide when the drought starts, which means that we can be well poised to capture the full extent of the drought across the entire ecosystem. So a really unique aspect of this study is that we are measuring everything in this ecosystem. We're measuring the atmosphere, the leaves, the trunks of the trees, the roots, the soils, and the water throughout the entire system. So we're going to have a really complete picture of how this entire ecosystem is responding to drought. Plants release thousands of compounds into the atmosphere, and we're going to track these with high-precision gas analyzers. And because of this, our team of scientists need to restrict the type of lotions and fragrances, perfumes that we wear, because these human fragrances can interfere with the type of plant fragrances that we are trying to track. And for the next few months, this is the reason that the rainforest will be restricted to scientists only, who are being very careful about the human fragrances that are released to the system. But we're very excited to share the results that we find from this study with you in the future.
Here at the Landscape Evolutionary Observatory, or LEO, researchers are studying a landscape in its first phase of evolution. LEO is an experiment to study the interacting systems of air, water, soil, plants, and microbes. It consists of 3 enormous steel structures filled with 1 million pounds of crushed basalt rock from a volcanic crater in northern Arizona. The forming soil in each structure is filled with almost 2,000 sensors so that scientists can measure the effects of their experiments on water, carbon, energy, and other cycles. By controlling different elements of this complex system, they hope to find answers to many real-world questions, including the effects landscapes have on the amount and quality of water runoff.
This past winter, we implemented a 45-day rain experiment on all 3 of our hill slopes. That water percolates through the soil and interacts with the thousands of sensors we have embedded at different depths.
Those sensors allow researchers to create 3D models of their landscapes, which yield unparalleled insight into the water cycle and how it relates to hydrology and ecology on our hill slopes and mountains. As a massive experiment, LEO will further our understanding of how the interactions between water, minerals, microbes, and vascular plants help sustain life on Earth.
When the biosphere was first constructed, the idea was that it would have a representative biome from all around the world, and of course the ocean, being 76% of the Earth's surface, was included in that. The ocean as a system itself is developed to be able to accommodate a pretty complex ecosystem. It's not just an empty pool. So there's a deep end that goes down to about 27 feet. There's a shallow part in the middle that comes up, and then there's a shallow area, like a beach area. We've got a wave machine. During the school year, we bring in classes from schools all over the region to learn a little bit about marine science. So we can tell them, for example, about how climate change impacts the ocean. What is ocean acidification? The fact that the CO2 we put in the atmosphere is actually taken up by the ocean and changes the chemistry of the ocean, and how that has detrimental impacts on ocean life throughout the ocean. So even though we live in the desert here in Arizona, it's important to understand the role that the ocean plays in our daily lives.
Important, too, is to gain a sense of how individual ecosystems contribute to global systems of climate regulation, water cycling, and soil development. Earth science has revealed that human beings are a significant agent of change on our ecosystems. We ourselves are changing the way that systems on Earth interact and evolve. Because of this, the human factor must play an important role in our scientific studies. Humans were part of the first experiments conducted at Biosphere 2. Between 1991 and 1994, there were 2 missions whose purpose was to study how to create a closed ecological system with the ability to support human life in outer space. 8 people were sealed inside Biosphere 2 for 2 years. One of their goals was to learn how to grow all of their own food in a way that was sustainable, using limited resources of land and water. To do this, they farmed agricultural plots in the part of Biosphere 2 that LEO occupies today. Biosphere 2 is no longer a closed, sealed system. Now, the 40-acre Biosphere 2 campus is being used as a model city to develop and test technologies for sustainable power sources and better resource management.
As population increases and climate changes, the relationship between food safety and security, energy and water becomes more and more critical. There will be places that will have less water, places that will have more energy or less energy, and food is going to be, in the end, critical. So at the Biosphere 2, through the years to come, we will be the place to evaluate the best technologies that exist in the nexus of water, food, and energy.
Biosphere 2 is focused on answers. We are simultaneously learning about our planet's past while forecasting its future, understanding our natural resources while engineering long-term alternatives. Biosphere 2 is an introduction to the adventure of scientific experimentation and to some of the largest forces at work on our planet. It's also a celebration of the remarkable biodiversity found on Earth. It's a place where scientists ask some of their biggest questions to get answers that benefit all of us. Biosphere 2.
So my name is John Adams. I'm Deputy Director here at Biosphere 2, and we're standing inside Biosphere 2's desert. Just like the desert, and you can see how large the space is, Biosphere is a remarkable research facility because of its scale. There's no other place in the world that you can do the types of earth and environmental science projects that the University of Arizona does here. We're studying how does water move from the mountaintops into the aquifer and what impacts that quality, how are coral reefs in the oceans going to respond, and how are our rainforests going to respond to temperature and drought. Hello, my name is John Adams, and I'm Deputy Director here at Biosphere 2. We're inside Biosphere 2's coastal fog desert. This type of a desert is different from that which we find here in the Tucson area. And so if you were to travel from southern Arizona down the Baja coast, you would encounter many of the types of plants that were incorporated into this biome. The unique thing is that you travel through several different desert regions in and of itself. And in this system, we've packed together plants that would typically not be found assembled together. For example, we have Joshua trees, we have cordones, we have bujums, and again, some of these specimens would never be found in such close proximity to one another, but because you're able to recreate ecosystems inside Biosphere 2, the designers were able to achieve this. My name is John Adams. I'm Deputy Director here at Biosphere 2. One of the really unique and institutional experiments that we have going on at Biosphere 2 under the management and ownership of the University of Arizona is LEO, the Landscape Evolution Observatory. We've constructed and completely repurposed the former agricultural space to now provide us an opportunity to better understand it rains in the mountains, how much water ends up downstream for you and I to use, and what impacts that quality. And we've had some really interesting finding because hydrologists will tell you that most of their models were set up and designed and the algorithms that are used in the 19th and 20th century. We have very little ways to test the validity of those. Well, LEO offers them a new tool for them to better understand those dynamics and hopefully be better predictors as what's happening to the hydrologic cycle outside. My name is John Adams. I'm Deputy Director here in Biosphere 2, and one of the really unique features of Biosphere 2 is our equatorial rainforest. Now, although it's not a perfect representation of that which you'd find down around Brazil, it is an assemblage of tropical plants inside of a completely controlled environment. What makes this unique is we've got plants that are only a couple of feet tall to plants that are upwards of 80 or 90 feet in height and have been maturing in this facility for nearly 25 years. This allows our researchers to make changes to look at how this entire system responds. And when we see a response, we have a much higher level of confidence that the response we see is attributed to the change and not that the fact that the system is going through some level of stabilization. Some of the research we're doing is it's predicted to get warmer and drier in the tropics. Well, how do you turn up the temperature or turn off the rain in an actual rainforest? You can't. Inside Biosphere, we can do that, and we can get a much better idea of the adaptive strategies these plants have in dealing with these extreme conditions. Hello, my name is John Adams, and I'm Deputy Director here at Biosphere 2. One of the truly unique features of Biosphere 2 is we have a 1 million-gallon ocean. It was originally designed after a Caribbean reef environment, and early on, some of the most significant research came out of this area. And we were able to show a direct connection between increasing atmospheric CO2 and decreasing coral calcification. And what I mean by that is that the ability of corals to grow was significantly impacted in a negative way as CO2 in the atmosphere goes up. Today, we have a really unique opportunity as we have a group of researchers from around the world who are interested in repurposing our ocean to continue not only a better understanding of increasing CO2 and its impact on corals, but also how is that further compounded by the warming conditions that we see around reefs.
We're going to talk about what is a coral. Now, corals are an animal that actually live in this calcium carbonate shell. They live in these small little pores of this shell here. And they have this really unique symbiotic relationship with something called a zooxanthellae, which is a small plant cell that lives inside of the tissues of a coral. Now, that's important because corals actually rely on those zooxanthellae for their energy. That photosynthesizing process provides them with the energy that they need to survive. So coral is an animal. It lives in marine systems and is most commonly found in coral reef environments. Corals are incredibly important for a couple different reasons. They're a place that 25% of the marine biodiversity that lives in our ocean, they depend on coral reefs, whether it's for a place to live, a place to be sheltered, or a place to find food. So that's a large amount of animals that depend on corals. Now, we as humans also depend on corals because fish depend on corals. We depend on those reefs being stable in order to continue to have food. The other reason corals are important is they're a huge part of economic development for a lot of countries. Countries rely on ecotourism. They rely on coral reefs, bringing people out to their nations and to their different places that are coastal environments. People go on vacation to coral reefs because that's what they want to see. So a lot of people rely on coral reefs for ecotourism. So inside of our Biosphere 2 ocean, we have some really cool invertebrates that are living in here right now. This that I have right here is a black long spine urchin. These guys are known for being voracious herbivores. They love algae, so they eat it at a very rapid rate. So we're just now in a trial period with these guys to see how they do, and they're doing really well. One of the things that we're trying to do in this ocean right now is decrease our levels of algae. So we have about 15,000 hermit crabs. So we have about 25 of these black long spine urchins. We also have about 20 strawberry crabs, and all those animals help us clean up this algae. They eat it really quickly. So we're doing that right now to try to decrease this algae in preparation for this coral project that we'll be starting. So this black long spine urchin is a really important part of our Biosphere 2 ecosystem out here. One of our other invertebrates we have inside of our Biosphere 2 ocean, along with the hermit crabs and the urchins, are these really cool little strawberry crabs. So this strawberry crab also loves algae. Now these guys have a really cool adaptation. So you can tell that they're bright red. Something that they have in order to kind of deal with that is they have these small little hair follicles you can barely see onto the top of its shell. Now those little follicles actually help collect algae. So you can see that it's got some green on its top. So that's a huge part of its camouflage method. They don't have to worry about worry about how red they are so they don't get preyed upon. Now that's not really an issue in our Biosphere 2 ocean. We don't have anything that's gonna eat these guys. They've got a pretty great life. All they have to do is go around and they can eat all the algae they want and get to just graze in our Biosphere 2 ocean. So coral reefs, as cool of an animal as they are, react really strongly to different stressors. And we've known that about coral for quite some time. We started to see, as carbon dioxide increased in our atmosphere, we started to see coral having an invert reaction to that. They started to actually, their shells started to disintegrate because the ocean was becoming more and more acidic. The other stressor that's coming in now is that our oceans are getting warmer. They're getting warmer by 2 degrees Celsius in a lot of places. A lot of corals don't do well with that increase. Any kind of change in their environment really adds a lot of stress to their plate, and they're not able to grow or survive. They also have reactions where they actually expel their zooxanthellae, that symbiotic plant cell that lives in their tissue. And if they don't have that, then they're having to find their own energy. So these corals being under stress is a big problem. We've lost 30% of our Great Barrier Reef in 2016. So our coral health is going down, and we're losing corals at a rapid rate, to the point where we need to do something about it, and we need to do something about it quickly. So at Biosphere 2, we are working with coral researchers from around the world, from the University of Hawaii, University of Miami, all different places, University of Australia, and we're looking at how we can prevent corals from leaving our oceans. So what they're doing is they're actually selectively breeding the corals that are surviving this increased temperature, the ones that are more resistant to these stressors. When they selectively breed those corals, they're making a super coral, one that's even stronger than its parents. That super coral is what is going to be surviving in our oceans. They're doing these tests in the laboratories now in Hawaii, and we're hoping this is going to be phase 2. We'll actually bring those super corals out to the Biosphere 2 ocean and continue to do testing on them before we do anything in the natural systems. Out here at Biosphere 2, we are working with coral researchers from all over the world, from the University of Hawaii, University of Miami, even some
Good morning, everyone. My name is John Adams, and I'm Deputy Director here at Biosphere 2. I'm really excited this morning to show you and take you on this virtual run of the historic Biosphere 2 property. Now, although you can't be with us here today, we really hope that you'll be able to join us in the fall or early spring when we will be able to reschedule this. So stay tuned to Run Tucson's website, and they will provide all the necessary information. Well, why don't you go ahead and join me now, and we'll go through.
Crossbreeding them together to create a super coral. So they're essentially speeding up evolutionary processes to create this super coral that will withstand the increased temperature. Now, we're going to be doing that in our Biosphere 2 ocean right behind me here. We'll be putting those corals inside of this ocean and being able to do different modifications to it, different testing to see how these corals are reacting. So they're doing it in phase 1 in the laboratories now with just these little coral fragments.
This is a great place to just sort of take a stop because you're running by the historic B2 Ranch. We call this the Ranch House. It dates back to the 1920s, the actual original homestead by a dentist in Tucson by the name of Dr. Lackner. And so he had a small cattle ranch and used this as a hunting lodge at that time. So this course offers just some spectacular scenery. Over here to the right, you have the Canadalo drainage with the Catalinas rising up. I think some of the most spectacular scenery in all of Tucson. And then all you have to do is look over to your left and you've got Biosphere 2, and this is really a unique angle that most people don't see. Well, welcome to the historic Suffolk House on the Biosphere 2 property. This is another historical building that you're going to have an opportunity to run by on this virtual EarthFest. And it dates back to the 60s when a person by the name of Lady Margaret of Suffolk built this home. She thought that Tucson was encroaching on her, and so she moved out here and built this estate. Okay, you've made it. This is the finish line, and this is where all of our runners will end when we're able to hold this race live. Now, We hope you really enjoyed us sharing with you this spectacular campus with you, the scenery that's here and the historic buildings that make up Biosphere 2's site, because many people don't think of the property having this historical value, but it does, in addition to the cutting edge research that's going on inside Biosphere 2. So we hope you're all staying safe, and again, thank you for allowing us to share this unique property with you today. Good morning, everyone. My name is John Adams, and I'm Deputy Director here at Biosphere 2.
Out here at Biosphere 2, we are working with coral researchers from all over the world, from the University of Hawaii, University of Miami, even some universities in Australia. And we are collaborating with them to figure out what we can do to save the coral reefs. Now, they're doing some preliminary testing. They've started doing some testing in the University of Hawaii laboratories, where they're taking corals that are more resistant to this increased stress, the increased heat that we're seeing in our oceans. The ones that are surviving this increased heat, they're actually taking them and selectively breeding them, crossbreeding them together to create a super coral. So they're essentially speeding up evolutionary processes to create this super coral that will withstand the increased temperature. Now, we're going to be doing that in our Biosphere 2 ocean right behind me here. We'll be putting those corals inside of this ocean and being able to do different modifications to it, different testing to see how these corals are reacting. So they're doing it in phase 1 in the laboratories now with just these little coral fragments. We'll be having more of a large reef system put into this Biosphere 2 ocean to even elaborate on the research even more before we try it in the natural systems.
Good morning, everyone. My name is John Adams, and I'm Deputy Director here at Biosphere 2. I'm really excited this morning to show you and take you on this virtual run of the historic
With climate change, shifting weather patterns around the world, and fires
everyone, welcome to our Facebook Live. My name is Katie Morgan, I am Manager of Marine Systems, and today we're coming to you live from our Biosphere 2 mangroves. This system has been around since the original crew members were inside of Biosphere 2. These mangrove species, we have 3 species of mangroves. We have black mangroves, red mangroves, and white mangroves. These mangroves were brought in actually from the Everglades all the way over to Arizona in some trucks. That was quite an adventure as we understand it. They actually got stuck at the border of Arizona trying to get these plants in because Customs didn't have mangroves on the list of species that could be let into Arizona. So they kind of got stuck there for a little while until they got the correct permits and permissions to bring them over here to Biosphere 2. And then they were quarantined, these mangroves were quarantined until the space was ready for them to be put in. And the unique thing about this biome is that not only were these trees brought in, but so was the water and the soil and the microbiome that comes along with that soil and water. So it's one of the most full biomes that we have out here. It all came from one place right here in front of us here. And you can tell the difference between these trees. So right off over here are red mangroves. You can tell because they've got these more aerial roots. So these roots are higher off from that water's surface. And then over on the other side are our black mangroves. You can tell those roots are different. They look kind of like fingers emerging from the water there. So those are how you can identify the differences between those 2 species of trees. And like I said, we also have some white mangroves in here. Those white mangroves are on the other side of this marsh mangrove system. So you can't see them from the perspective that we're at, but they are. There's a couple trees that are on that side. Now, when these trees were brought in, none of these trees were taller than 5 feet. So you can tell there's been quite a bit of growth in the 30 years of this system being here. These trees have done really well in this system. Mangrove environments in general are incredibly resilient ecosystems. So the water that's surrounding us right now is what we call brackish water. So it's a combination of salt water and fresh water, and its salinity is quite a bit lower than the oceans, but it's kind of that middle ground between the fresh and the salt water. So these trees are actually living in salt water right now, which is a very unique thing. Not a lot of trees or plants in general can do that. So they're able to adapt to this saltwater environment by actually pulling that water into their roots, and they extract the salt on the back of their leaves. So they don't use that salt for any purpose. They actually just go ahead and get it out of their system, and they're able to extract it on the back side of their leaves. Now, originally, when this mangrove system was put together, the idea is that we would have a freshwater marsh that would connect to the saltwater marsh that then would connect to our ocean system. Now, this makes a lot of sense when you think about it because the oceans are connected to mangroves and the natural systems around the world. But when you think about the scale of Biosphere 2, our mangrove system scale to our ocean system scales are not different enough for that to work. But they didn't know that yet until they tried it. So they actually did connect these systems for a very short amount of time until they quickly realized that the nutrients in the mangroves was way too high to be putting into the ocean for it to be successful. So these systems are completely separate today. Our ocean and our mangroves do not have any interaction, but they did in the early 90s for a very short amount of time. Now, there's a lot of research that has come out of this ecosystem here. There's been research looking at the differences between a system in a closed environment, like we are inside of Biosphere 2, compared to those trees that are out in the natural systems in the Everglades. We've looked at different things, including leaf litter. So how does leaf litter decompose in this kind of system? You can see a lot of the leaf litter down below us here. So they looked at those decomposition rates in the leaves. But they've also looked at levels of insects and insects eating and destroying some of these plants. Now we have very little insect life in here that is negatively impacting these mangroves. So they've been able to do studies looking at how the mangroves in Florida are reacting, having stress reactions, and how that might affect the anatomy of the plant, versus our mangroves in here, which have very little insects that are destroying their system. We have a unique collaboration with Patrick Kangas from the University of Maryland, and he is actually, for many years, he's been involved with the project since day one. He helped actually design this system, but he's been bringing out students from the University of Maryland to work with us here and has built an algae turf scrubber system. Now, algae turf scrubbers have been around for a long time. It's actually how we controlled the nutrients in the ocean initially in the first experiment. We've moved on to some more modern technology today, but algae turf systems are really beneficial for areas that have high nutrient loading. So mangroves are a really great example of a system that has really high nutrients. And in order to kind of extract some of those high levels of nutrients to increase productivity, they're able to use these algae turf scrubbers to grow the algae, and then you pull that algae off of it, kind of remove some of those excess nutrients to keep the system healthy. And then those algae that's being pulled is being looked at for things like biofuel and food sources. So there's a lot of innovative research going on looking at how to use mangrove systems and technology like these algae turf scrubbers to think about how we are better in the future. Now, a lot of people ask when we're in here, what kind of animals do you have in here? I think that's a common question we get throughout all of our biomes. Now, I mentioned that they brought in soil and water and the microbiome from Florida, and that is true. Along with those we have two species of fish in here, a killifish and a mosquito fish. The main fish that we have in here today is our killifish. We don't see many of those mosquito fish in here anymore, but we do have those swimming throughout. But we don't have a lot of the other traditional animals that you might see in mangroves, like fiddler crabs or sponges, or any kind of like a lot of juvenile animals. So mangroves are around the world very beneficial for a lot of our juvenile marine animals. A lot of fish and invertebrates actually go and lay their eggs amongst these roots. You can see how protected these roots may be. Those animals are coming in and burrowing in, laying those eggs. It allows a protective area for these animals until they are big enough and strong enough to go back out into the natural systems. Now, mangroves also play a couple other really key roles around our world today. You can tell these root structures are all up in this soil, so they actually are really great at preventing erosion. So having these complex root structures keeps all that soil in place. And if you think about it, where do you find mangroves? Well, you find them along our shorelines. And so they're really important for when we get big storms, hurricanes. They keep our shores intact. Now, this has been an issue as we think about developing our coastlines. I mean, most people, when they go on vacation, they have an option to stay right on the beach or 2 miles away from the beach. They're most likely going to want to stay right on the beach, at least I would. So a lot of our mangrove systems have been destroyed because of that, because of development. And we've really been able to put in protective measures to make sure that no longer happens, because not only do these mangroves protect our land, keep it from erosion, and protect those baby marine animals, they also are really great filters for our ocean. So these roots take up tons of excess nutrients, whether it's from pollution or just excess things that are coming from the ocean, and filter all those things out so that the oceans are cleaner and our mangroves are cleaner. So there's so many important things that these mangroves do, and we're lucky to have a system like this inside of Biosphere 2 that we can continue to use for research and compare a system that's been under glass for almost 30 years versus our natural systems, which are looking quite a bit different today. So it's a really unique research opportunity for us today. So thank you so much for joining us. It was a pleasure to bring you into the mangroves today. I do want to give a shout out. We are going to be doing a virtual Earth Day event. Earth Day is coming up on April 22nd. So keep your eyes peeled. We're going to be posting some information about that early next week. So we hope that you're able to join us for that. And we'll see you live from Biosphere 2 next week.
Good afternoon, everyone. My name is John Adams. I'm Deputy Director here at Biosphere 2, and I'm welcoming you to our daily live session on our social media platforms. And today we've got a real treat for you. Biosphere 2 has two large geodesic domes that are called the lungs. And they're probably one of the most remarkable engineering features of Biosphere 2. Now remember, Biosphere 2 was originally designed as a completely sealed environment. No material exchange, and the only thing that would move in and out of Biosphere 2 under its original design would be energy. Now what happens when you have a large mass of air inside of a fixed volume and you heat it up? Well, what happens is that means the pressure goes up. And conversely, when you cool that air mass down inside, then the pressure goes down. Well, the engineers quickly determined that if they didn't account for these changes in pressure, you could compromise the integrity of Biosphere 2. It could explode, or it could implode. And so what we're going to do is we're going to walk you down into the lung. But first, what we're going to do is to get to the lung, we've got to travel through a tunnel. We're going to walk you down it. This tunnel, and there are two of these, the one we're going to go in particularly goes underneath the road that connects the desert and the lung. And so why don't you go ahead and follow me down the tunnel, and then we'll go into the lung chamber itself. So this tunnel is pretty cool. It's got some great acoustics. When we did have school children here, this was probably one of their favorite places. They would really like to let out some noises because the reverberation is overwhelming. But, you know, the Biosphereans, when they originally designed Biosphere 2, thought of everything. So, not only would air travel in and out of this tunnel, but we're also, as you can see, on a downward slope. And this slope starts in the rainforest and works its way all the way down to here. So, if you're inside Biosphere 2, and one of your water pipes breaks, you don't want that water just running any place. You want to be able to try to collect it. So everything would work its way down into the lung where we have a large reservoir to collect all that water as well. So it's really ingenious how this particular space has many different uses. So as we go through here, you got to watch your step in your head. So let me turn on some lights in here. You can actually get an idea of just how big this chamber is. So, I mean, I think the most common reaction we get from people in here is, wow, and how cool it is. I mean, it just looks like something out of this world.
With climate change shifting weather patterns around the world and fires continuing to rage in Australia and the Amazon, the question of how carbon gets stored, used, and released in the environment is getting more attention. Researchers from over 80 institutions are collaborating on a project to track how the carbon flows in a rainforest under drought condition and as it recovers. But it's not happening down in the Amazon, it's happening in Arizona. You know, the enclosed dome of the Biosphere 2 habitat? They're letting researchers use things like carbon-13 labeled CO2 to follow the flow of carbon through the air, the plants, and the soil.
One of the cool things about an enclosed ecosystem like the Biosphere 2 is that you can control the weather. So scientists decided, why not turn off the rain for 2 months and force this rainforest to go through a drought? And one of the big questions they have is what the plants are going to do with their carbon when they're stressed out.
You guys are measuring VOCs, which are volatile organic compounds. And from what I understand, these plants emit these compounds that sometimes even have fragrances. Connecting it back to humans, it almost feels like we're measuring the sweat of plants.
You can even measure human sweat, has a lot of volatiles. There are people who are measuring like when people are in stress, we also start to emit a lot. It's not just only the plants, it's quite interesting. And it seems it's not only the plants, it can be the soils, it can be the microbes, and there's not that much known, so we really monitor everything. What we're setting up here is a measuring system, everything from the leaves down to the soil, where we can actually monitor. So we can see how the drought kicks in, how deep it goes, how the species respond, and then start the rewetting whenever we think we have that extreme drought we want to see.
Inside the rainforest is this fake hollow mountain, and inside that mountain is a room that was filled with very precise scientific instruments. And there was this spider web of plastic tubing that connected those instruments to every part of the rainforest. It was something like 2 miles of tubes.
Wow.
And they were constantly collecting gas samples from different parts of the rainforest and figuring out where the carbon was going.
These gas analyzers are organized together on streams, measurement streams that we use to look at the atmosphere, the leaves, the tree trunks, the soils, the roots, many different components of the ecosystem simultaneously.
So we know that forests soak up a huge amount of carbon, but we don't know exactly where that carbon goes. And that's because in the real world, you just can't track carbon cycles that closely. But the great thing about the biosphere, too, is that it's a sealed ecosystem. So one of the experiments the scientists did is they closed all of the doors and they shut themselves inside for 4 hours. And I got to be in there with them. It was a great deal of fun. And they released canisters of carbon-13 into the air. So that's heavy carbon, and it's something they can track with scientific instruments. And then they had these instruments hooked up to every part of the rainforest to see exactly where those carbon molecules ended up.
We're watching to see the whole ecosystem draw down that 13 carbon as plants photosynthesize the CO2, fix the carbon, and then use that carbon to produce biomass, to produce VOCs, and do all sorts of things that we're tracking simultaneously. Since we added this unnatural amount of 13 carbon, we see a really large enrichment, and that's what this pulse represents. We open the tanks outside and let the CO2 flow into the atmosphere. It drove the 13 CO2 ratio above 1,000 per mil, which was our objective. And then we saw this steady decrease over time, and this represents the amount of that 13 carbon label that the plants took up during photosynthesis. We did this for over 4 hours, opened up the glass windows and flushed the system really fast to try to get this 13 carbon label out of the atmosphere so we could start to look for 13 carbon in CO2 that re-emerged because plants photosynthesized it, they fixed it, and then it got re-released from the ecosystem.
You know, if you're studying climate change and you're looking at ecosystems in the real world, they're very complicated and it's super hard to control all of the variables. But if you're just looking at like a potted plant or a greenhouse, well, that's pretty small and you're not really getting the complete picture. So this rainforest is the size of a gymnasium. It's been living and thriving for 30 years. It's the perfect place to do this kind of research.
So now we really see strong differences already. It was very species specific, so some species like these big clitoria trees here, they started to shed leaves early on, so it looks pretty much like autumn. Yellow leaves are falling down. Other plants hang in much longer, didn't show any visible signs, but we can see it in all our measurements. And now the soils really got very, very dry. We have lots of falling leaves, and now we're at this point where the drought is really severe, so we see leaves hanging down. Some of the plants start to look pretty bad, pretty yellowish. We won't let anything die here, but we are at the extreme drought point now.
You guys with papers and virtual EGU, is that happening?
Yeah. Well, these this is then the baseline, which can feed in anything. And the one thing we are, for example, monitoring is the volatile organic compounds. These are small little molecules which the plants are emitting, which actually give us the smell of the forest. And the interesting part is we can see in the moment that they're actually uptaking.
We're actually going to take some time that week and do a writing workshop, so work on the paper specifically. That we do that like in person in Austria, but that's what we'll do remotely this year.
It's amazing. And it's really interesting to see how this is happening. So at the moment, the main part comes from the leaves. But further on in the next days, we wonder how it goes.
Laura and Christiane, we're just going to wait a few more minutes for folks to come in.
Contributor. At the moment, the soils can take up also the atmospheric molecules, but then it will go through the roots, the carbon fed into the microbes, and they might also release or take up other VOCs.
Her PhD from the University of Belfield in experimental biology and ecosystem studies. So it's a real privilege to have the two leaders of the Biosphere 2 WALD, which stands for Water, Atmosphere, and Life Dynamics, the Biosphere 2 WALD campaign, which took place this last year in the rainforest. We have those leaders here with us today to answer some questions. And I want to start off in the spirit of Earth Day, and I'll let both of you answer. This will be the hardest question of the panel. What is a forest? And Laura, let's start with you.
Well, one definition of a forest could be a type of ecosystem that invests a lot in reaching up above ground to extend the way that plants interact with their environment to higher and higher levels in the canopy. And so really when we think about forests, I think we think of the extension of competition for light upward into the atmosphere. And it's just another illustration of how strongly the biosphere and its plants and animals and microorganisms are interacting with our atmosphere.
And Christiane, how about your view of what is a forest?
Well, here in Germany, there is a saying, a forest is more than just a lot of trees. And I think this is pretty much assembling it. It's a very complex interplay. Of course, we have the dominant tree canopies, which build up this forest. But in the end, the forest is everything. It's the soil with all its processes, the microbes, the roots, the mycorrhiza. It's the trees, it's the animals, and it's a lot of exchange happening within this system. So it's a quite interesting and very complex system. And I think looking worldwide, these are the biggest ecosystems taking up carbon. So they're really important to our forests. And that's why they are so interesting to study.
Now, forests around the world are in a fairly abundant supply, but why did you two choose a forest inside a greenhouse system out of all the forests out there to do your research?
Christiana, do you want to start with that? Because you had the idea even before I did.
Yeah, I think the forest and the biosphere is absolutely unique, because what we have is both. We have both like a controlled system and a natural system. And I think this is really the one place on Earth where we can control, manipulate, and measure a full ecosystem of a forest inside what we would call a big cuvette. So normally we study plants by enclosing a leaf or putting a single plant into a small cuvette. And here we have a full forest which has been grown very naturally for many, many years, and are able to study all these processes within an enclosure where we can control the atmosphere, we can control the rain, and many other factors. So for a scientific research program, this is just a unique forest where you have the possibility of big scale manipulation experiments.
And Laura, do you want to chime in on that?
Yeah, my research, I'm really interested in what happens below ground. What do soil microorganisms do to make their living? How do they survive? How do they compete with each other? And an important aspect is also how they interact with plants and interact with the environmental factors and forcings. And so already those are so many different interactions that it illustrates how complex it is to study soils and soil microbes. And so it's really a wonderful thing to have this extra level of control. For example, we turned off the rain so you can control whether or not it rains. And we also introduced different tracers, isotopic tracers, that allow us to sort of have more leverage to understand what is happening even in complex ecosystems. So for me, it's I think there's a lot of advantage to studying the above ground forest. And then with this extra control, you can also study the complex interplay with below ground processes.
And I think Laura just mentioned really the one big point for our experiment, and that was the ultimate reason why I choose to come to Biosphere 2 is we use this stable carbon isotope labels, which are just naturally abundant, so they're not radioactive, but we can apply them to give a signal in the atmosphere, and we can see how this is uptaken by plants and how this is traveling through the plants, through the trees, into the soil. And this is something which would never be possible in a natural forest, because if you would just blow it in the air, it would just go by the wind. So this is absolutely unique that we could close the whole system airtight. We could apply this big label study and see how the carbon is traveling through the system. And that was actually the first idea when I went to Biosphere 2, and I thought, like, well, this is the one place where we can do that.
And for those of you just joining us, we are talking about a rainforest enclosed in a giant greenhouse. And this greenhouse is a part of the Biosphere 2 facility in southern Arizona. And within this rainforest, there are a variety of kind of sub ecosystems. And maybe Christian and Laura, you could just kind of give a blanket description of your rainforest. How many trees, plants, what kinds of features? Everybody right now is looking at the rainforest mountain, but maybe just take us into the forest in your own words.
Mm-hmm. So the forest is beautiful because it's heterogeneous. That is, it has a lot of different microenvironments within it, like real ecosystems do. And so this is one other advantage beyond going to, say, an agricultural greenhouse to study monocultures of crops. In this system, we have about 70 different plant species now, and multiple different types of trees that are dominating the canopy, and they're living in different microenvironments that have different miniature watersheds, different elevations, which translates to different environmental forcings. And this is really great for us because we know that in real ecosystems, this type of complexity and spatial differences really do impact the whole ecosystem process. And that's something that we can't study in monoculture. And so it's nice that we have this extra level of control that we were just talking about that helps us simplify some of the science, but yet we're not simplifying how an ecosystem can be complex. So up and down this mountain you see in the middle, we have different clusters of different plants in lowlands, in floodable forest areas, in the upper layers that are very, very hot, that really allow us to look at diverse ecosystem processes and diverse responses of plants and microbes within this ecosystem.
And just a little bit of context with a question from Facebook. Somebody asked, why didn't you include animals in the biosphere? And perhaps you could speak to that question, but also speak to the fact that this forest that you're studying is about 30 years old. And it was designed 30 years ago with tons of species, including some species and some animals, and excluding some species and some animals.
Mm-hmm. Yeah, I can take that again. So, I mean, these are engineered ecosystems from the beginning. And one of the concepts was to overstock with different species and allow some of the natural selection processes to happen to ensure that there would be survival, but some of the best suited plants and animals for that ecosystem would survive. And initially there were a lot of different animals, different members of what we call the trophic levels, so like different members of the food chain that were present in this ecosystem. And of course, there was a lot of effort that went into making sure that none of the plants or animals would be dangerous for the humans that were living inside at the time. Today, we really mostly have plants, we have microbes, and they have interactions that have developed over 30 years, which is really great. There are some insect species, but the main animals in there are humans, our scientists, and our tourists that we really enjoy showing the facility when we can. And that is not the same as a tropical forest that will have really high biodiversity and a lot of animal life. But for us, we're mainly studying carbon cycling, water cycling, and actually a lot of our ecosystem models, they don't necessarily include animals either. And so we're still able to test some of the most fundamental understanding we have about ecosystem carbon and water cycling with this model system, even though we're not necessarily representing some animal interactions, which of course are important, but we feel we have a lot of other things to learn in the meantime, even without them.
Yeah, and kind of playing off of that, we have a question from Norway. Rune asked, how are rainforests worldwide being affected by climate change? And maybe you guys could connect that back to your research once you answer it.
Yeah, so I think worldwide there is a large concern about the carbon sink capacity, especially of tropical rainforests, declining. And there's actually a brand new scientific paper in Nature, which is extrapolating changes they have been observing in both African and Southern American rainforests, especially the Amazon. And it seems like the overall capacity to withstand these extreme climatic events is declining. And if these projections are right, the big carbon sink, which currently these tropical rainforests are providing, they're kind of the lungs of our planet, absorbing a lot of CO2. This might decline with global change. Severe frequent droughts and recurrent droughts, and that is a phenomenon we unfortunately see more and more frequently. Droughts have always existed, also in tropical forests, but the frequency and the severity with which they are appearing is increasing. So that's also one of the reasons why we choose to implement a big drought experiment into this biosphere rainforest to really study under semi-controlled conditions how such a severe drought can impact these systems.
Thank you. And just to briefly describe, Laura and Christian spearheaded a research campaign this last year that consisted of about 100 researchers from all different fields. And in the Biosphere 2 rainforest, they initiated a severe full ecosystem drought. And that meant shutting off the water and letting the soil and the plants and the trees really dry out. And I will cut to a photo of a difference between a healthy forest and a forest under drought in this photo you see here. And maybe you guys could describe how you were studying this drought and maybe the different phases of experimentation, starting with a healthy forest versus forest under severe drought and stress.
Yeah, so we outlined it was about 5 months in the end, a 5-month experiment to understand how carbon and water cycling were occurring in this forest under control conditions. So on that photo you're showing on the top where the forest is healthy and happy and well watered, into a severe drought, we went for 66 days with no rain at all to this ecosystem that's used to getting rain every 2 or 3 days. So it was really severe. We also made sure to purge any groundwater, so any sort of deep water that was in the system. And so this forest was very stressed out. We didn't push it beyond the point of recovery, but we got it to a very, very stressed level. And then we re-wet the system and watched the recovery. And I'll let Christiana describe some of the isotope labeling, but just briefly, we, in bringing together this really large group of scientists, one of our goals was to consider many different parts of the ecosystem that were responding together so that we could attribute if we saw some kind of carbon pulse, was it coming from a leaf process or a stem process or the roots or the soils? And so we built an online measurement system that was measuring over 130 points within the ecosystem all the time. And we were doing that with kilometers of this Teflon tubing that you see in the photo, extending out to leaf chambers or root chambers or stem chambers or soil probes that would let us look at all different parts of the ecosystem. And we had an instrument shed that housed our specialized instrument to measure carbon dioxide, water, and their isotopes. And we're even looking at these volatile organic compounds in the air. They're like the fragrant, low abundance, but fragrant compounds that we can smell with our noses that are really important to plant defenses and stress mitigation and also to microbes. And so we had a central shed where we had a lot of instruments that were connected out like in a spider web to the forest to kind of take the pulse of the ecosystem as we took it through this drought and then the recovery phase. And alongside that, there was a lot of other type of sampling that was happening simultaneously. This photo here is showing Eric Daber, one of our graduate student scientists, and he was working on that root chamber. And then afterwards, we would take a root sample, for example, store that and use imaging technologies later to better understand it. We took a lot of soil samples, leaf samples. So it was sort of this very integrated experiment looking at many different facets of the ecosystem. And so we started with this beautiful shed that we received a generous donation from Daniel and Susan Warmack, to construct and to power and to cool. And we filled it with a lot of dollars and euros of instrumentation, as you see on the bottom right, that we received in shipments from our colleagues from Europe and in the US, and also brought instrumentation from U of A. So yeah, it was a very comprehensive system for monitoring the ecosystem.
And if you're just joining us, because we have had some attendees dropping in mid-webinar here, we're honored to have Christiane Werner and Laura Meredith joining us. Laura is an assistant professor at the School of Natural Resources at the University of Arizona, and Christiane is the chair of ecosystem physiology at the University of Freiburg in Germany. And we've been getting some questions from all of our listeners this morning. And Robert Christofferson had a question about the instrument shed. And now that the instrument shed, now that your experiment is no longer in active research, what does the experiment shed look like now? And maybe go into the process too of having to ship all of this equipment across several countries to get it built in the first place.
Yeah, well, Christiana, would you like to start with the building phase?
The building phase was a nice phase. It was amazing. We had lots of support from the biosphere. It's something really uncommon that you can study something like a forest, but on the same time have really all the luxury of something like a lab next to you. But there were many, many challenges. The first thing was shipping. So lots of shippings got delayed. One of mine got fully broken, so weeks of repair. But we had this great support from the biosphere. Simple things like European instruments need different electricity than US instruments, tons of different plugs and stuff. It's sometimes you think you do this big scientific project, and in the end you struggle with a lot of small things to get this set up. So it was really weeks and weeks and weeks of plumbing, drawing these lines, having a very motivated team to build this all up. Yeah, I think without really all the support, we wouldn't have been able to get all this equipment in place.
Mm-hmm. Yeah, and from the photos, you can see how densely we packed. We had at least 20 different gas analyzers at one point in time in this room. And the takedown was, we had colleagues come back or who were there who helped disassemble and ship their instrumentation. And we also relied heavily on the University of Arizona team and the Biosphere 2 crew to help disassemble and ship back a lot of equipment. And so right now, the shed very much is emptied out and looks similar to kind of how it started. We've tried to keep it clean. We have some instrumentation that's staying, but really it's sort of now a blank slate for another research campaign to come in and ask new questions and build on what we're learning. We did invest in having more environmental sensors in the rainforest. So we have more soil sensors, more atmospheric sensors that are there and ready to use. So, but yeah, this was a campaign style type of research where we brought in a lot of equipment, focused very intensively to be able to characterize so much of the ecosystem at one time. And then we disassembled and we'll start over again someday. So
another great question we have from Tucson here. They're asking how the biosphere is used both for climate change studies, but also as a proxy for studying past climates. And this is an interesting question because we have researchers that are interested in all of our biomes at the biosphere of connecting what they see in real time by manipulating the biomes in the biosphere with events that have happened in the past. So for example, in the rainforest, how would a drought that you guys saw this year be related to a drought that may have happened 100,000 years ago in a rainforest somewhere? And what's the connection between those two studies?
Well, Cristiano, would you want to speak about Namaya Ladd's research? Namaya is pictured here in the pink shirt climbing in the space frame.
So Namaya is the third one of our leader team ship, and she is actually using biomarkers to study the past climate in plants. So there is a lot of information sequestered in, for example, leaf waxes, in compounds which are tightly linked to those volatile organic compounds which plants emit, which are also inside the leaves. And some of them are very, very stable for many, many thousand millions of years. So they can go into sediments. And what we can do is we can get information on the isotopic signature of these molecules and infer how the climate was in the past or how plants adapted to this climate in the past. And so this study we conducted here, this experimental drought, will also be used to see how this imprints its signature on these biomarkers. So we took leaves, Namaya will analyze them, or as far as we are possible to do it now with Corona. Of course, everything is standing still, but once we can work again, they will be analyzed in the labs. And so we have now an introduced climate shift with a long drought, and we had different phases of the drought. At the beginning, it was a mild drought, then we saw a severe drought, and we can see what signal this is imprinting on these biomarkers. And that can then help once we go to sediment cores or other climate records, compare these changes and see how, for example, past tropical forests reacted to different, for example, drought scenario.
Yeah, and there's a terminology from like satellite retrieval studies where people use satellites to take an image and do imagery of the Earth's surface or atmosphere. But an important part in satellite and remote sensing research is to ground truth, which means to do some work on the ground to verify that the image that you're getting is truly what you think it is, that your interpretation is correct. And in some ways, what we can do with this drought is to take samples that help us sort of ground truth, our interpretation of past climates or past droughts through things like isotopes in the sediment records, as Cristiano was just mentioning, or even in tree rings, different information that's stored inside of trees. And so one way to think about this controlled ecosystem research is a method to ground truth, our understanding in a model system.
Great. And we have a question from Heather. Do people still live in the biosphere? And I'll answer that briefly, and I'll just say that in the early 90s, Biosphere 2 was used as an enclosed experiment. And in the next hour, we actually have a talk with two of those original Biospherians, Jane Pointer and Tabor McCallum. So stay tuned for that. But nowadays, the Biosphere 2 is under different ownership in the University of Arizona. And it's being used to study enclosed ecosystems, particularly these model systems and mesocosms of rainforests, oceans, large hydrology catchment systems. And using the facility in this way has allowed us to develop these experiments that can give us some great insight, as Laura and Christiane are talking about, with things like grounding our understanding of climate change over large histories of time in truth, in scientific research, but also looking at other kinds of experimental interrelations between species in a forest. And I want to get back to your research because in this experiment that you guys conducted, you were studying a variety of both tree and plant species. And you noticed probably a fair amount of differences in the way that they respond to drought and stress. Can you share with us some of your findings about those interactions and whether they were interrelated with each other or mostly kind of individual dynamics that you guys were seeing?
Oh, maybe I can start. You have this nice picture up here, and this is one of the dominant trees, Clitoria. It's a nitrogen fixing trees. And what was super interesting that not just between the trees and the understory species, but also within the different trees, we found very, very different reactions. So what you can see already here is that tree on the early onset of drought started leaf shedding. So the leaves turned yellow, it looked a little bit like autumn, and it actually dropped a lot of its leaves, which is actually an adaptation for plants, because when you're getting into drought, if you have a large leaf area, you need to transpire a lot. If you can shed some of your leaves, the root surface to the leaf surface, the ratio is much better, and actually you can better supply your leaves still with water. However, that strategy only works in trees which have enough nutrients, enough nitrogen to regrow their leaves. So that's a nitrogen fixer is one strategy. And we found very, very different responses in the different tree species we were studying. And that was quite interesting to see the dynamics throughout the drought and how different trees responded at different times.
Yeah, and I'll say that one type of, we're looking at root processes and soil processes, and some of those analyses take a little bit longer. We took a lot of soil samples, and we preserved the microbial DNA and the microbial RNA. So the DNA is like the genes that they have, and the RNA represents the genes that they are expressing at that time because they need certain types of functions, like maybe they have a drought response function that they need to carry out during drought. And so we preserve those samples, and we'll be looking at a microbial species basis to see whether or not certain microbes were more or less active throughout the drought, and how that might be related to which plants they were interacting with at the root interface or even at the leaf interface. And so some of those data will be coming online as soon as we can get back in our labs and continue our research on those samples. But it's really nice to have complementary types of data, one where we're looking in real time at how carbon and water and VOCs are sampling with the gas analyzers, and then have other types of analyses that we do in the lab that help round out our picture of how this whole ecosystem is responding on a community scale, but also on kind of a species-specific scale.
And Robert Christofferson asks how the Wald experiment was different from any other drought experiments that have been done in the Biosphere 2 rainforest in the past. Maybe Laura, would you be able to answer that?
Yeah, I looked at some of the recorded past droughts that I could find in the literature and in our records, and this was actually one of, if not the most severe droughts that we've conducted. I wasn't quite expecting that, but 66 days is basically at that maximum threshold that's been carried out in the past for research purposes. So it was a very severe drought. We also were actively drying the air, drying the soil surface, so it was very severe. I know of past research that has added deuterium tracers to the rain. So this is an isotopic rain tracer, like they added heavy rain essentially and followed that through the ecosystem. And actually what we did in this study was the first way that we added water back to a very desiccated forest was from the bottom up. So we actually put heavy water, this isotopically labeled water, at the bottom of the ecosystem because there's a floor, there's a concrete floor, and then monitored the ability of deep-rooted plants to access that deep water and bring it up in their transpiration stream, and we're looking for signals of that in the soil system. And so in the past, there have been other droughts, and we've learned really interesting lessons about how inactive plants and microbes are during drought and how much activation they get when they're re-wetted. But we really applied a new approach to the isotope labeling this time. And I think in comparison to all research that has been conducted at Biosphere 2, we just had an unprecedented amount of sampling points and instruments and measuring, sort of monitoring at the same time, so we can have a more complete picture than we've ever had of how this system responds. And that was thanks to all these, wonderful folks we have. You see Christiana here in the photograph, along with Namaya Ladd, our other co-lead, and they're taking leaf samples, and preserving them very, very quickly by freezing them in liquid nitrogen so that they can later look at the different carbon pools in those leaves, including some of those volatile organic compounds that Christiana mentioned could even be trapped within the leaf itself.
Yeah, go ahead, Christian.
Yeah, maybe one thing to add. So the deep label watering, what Laura just mentioned, this is addressing a current really big research question: is how important are the deep-rooted trees for keeping water in the forest, for reaching deep water reserves? And that was just a unique opportunity to be able to actually add really at the groundwater level of this forest a labeled trace of it. The other really interesting and I think unique feature we have in this experiment is that we also labeled the CO2 in the atmosphere, as I mentioned in the beginning. And we did this before the drought and in the later phases of the drought. And what we could see already is there is a very close interaction between the carbon being uptaken by the leaves for photosynthesis, but then it's traveling down the trunk and it's going through its roots and directly ending up in the soil. And you see it coming out of the soil respiration again. So we traced all these pathways and how it's going into the microbes in the soil, these processes. And interestingly, once we applied the drought, all these processes were extremely slowed down. So already during the first phase, we applied the label in the leaves and we waited anxiously, and it took quite 2-3 days until we saw it in the stem, and then it took another 2-3 days until we saw it in the soil. And but on a drought, all these processes were way slower. So actually we're getting novel insights, not just what's the drought doing with the total ecosystem, how is the tree responding, but really how is this interaction between the vegetation component, between the soil component and back to the atmosphere is actually happening.
Yeah, one of our questions from Facebook was, where did all the carbon go? And you poked at that a little bit, but I just want to describe the effect to which you guys were studying these carbon pathways. And this research team with water, atmosphere, and life dynamics, they were setting up sampling systems at the level of leaves, at the level of tree trunks, in the subsoil. You guys saw some pictures earlier of actually people working below the soil surface in several soil pits, sampling soil and roots. And in addition, they were also measuring the atmosphere. So they created a real full systems view of how carbon that's put into the atmosphere starts to cycle. And maybe you guys can speak more to that process and that pathway, and how is carbon used by trees just in general?
It's a good question. I think I can start by saying that one really amazing feature of having what Christiana described as a forest in a cuvette, right? A forest in a contained system, is that we can really precisely measure the total exchange of carbon in and out of the ecosystem. And so we really know, how this forest is breathing to a very precise level. And so then our task is to drill down into the different levels within that to ask what are the different contribution from leaves, stems, root, soil within that. And so that's why we had this really extensive measurement system. And so what we can provide just with the sort of bulk measurements that we do is an inventory of this forest and where the carbon is coming in and out of and how much. And maybe I'll ask Christiana to describe how isotopes specifically help us look more deeply at how plants choose to spend their carbon and how they allocate it, and some questions we have in that arena.
Mm-hmm. So generally we can measure plant photosynthesis, which is the uptake of carbon already on the leaf level, or we can see the total drawdown in the ecosystem. But then once this carbon in form of sugars is in the leaves, there are many, many pathways of what the plant is doing with the carbon. So some of these are invested, for example, in stress protection, in compounds like these volatile compounds, which also the emission, especially we saw it for isoprene and later on in the drought also for monoterpenes, which are some of these specific VOCs, increased a lot under drought stress, which is a protective mechanism for the plant. But then most of the carbon doesn't stay in the leaf. It travels through the trunk. It can go into trunk growth. It can be stored. But a huge part of it is actually going below ground. That can be more than 30% of the carbon, which actually is going through the roots, into the roots, and from the roots to the microbes in the soil. And the nice thing when you use these carbon isotope labels is that you can actually trace it and follow it. So we had the analyzers, you can see it here with all these different lines connected to the trunk. We could actually see once this labeled signal of the carbon, which was fixed by the leaf, was traveling down and coming to the trunk and being respired by the trunk. And then we can see it in the roots. And in the end, when Laura will do all her analysis, we can see what went out from the roots into the soil microbes and was actually used by them. So this is the very nice way of these isotope labels, is that you can actually follow one tracer pulse you apply and how quick and how long it ends up. And in the end, we will also have tree rings and can actually see what is finally stored in the tree, for example, or in the roots or in the soil.
And we have a question again from Norway. How can this research contribute to actually saving the worldwide rainforest? And it's a great question because it kind of cracks it. How does scientific research fit into kind of the global narrative of some of our ecosystems that are at risk of natural resource extraction and changing climate and weather?
Yeah, I think part of what we're trying to understand here is the degree of resiliency of plants to stress, like drought. And tropical forests are not only facing drought stress, they face multiple stressors at the same time. And that's kind of increasing in the world as we see increasing frequency of droughts. There's also stressors from land conversion, let's say to a forest, to a different type of ecosystem, and then maybe regrowth to forest. So tropical forests are facing multiple stressors at one time. And so what we're doing here is very precisely controlling the drought stressor and asking how resilient are these plants and what are their mechanisms for resilience. And so one mechanism that Christiana mentioned is how much can they access deep water reserves, and can they sort of share that as a common good with other plants that are maybe not so deeply rooted? Or how can they allocate some of their carbon to help produce these volatile compounds that may help them mitigate some of the stress? And so I think one big aspect of what we're doing here is understanding the resiliency, understanding some sort of thresholds of these types of ecosystems that can then be put into our models that help us understand what this might mean on a larger scale, where we can't go in and make these detailed measurements everywhere around the ecosystem to assess that. We need to rely on our understanding that gets formalized in models to make those larger scale predictions. So some of it I really think is about understanding the environmental tolerances that these types of ecosystems have and their strategies for dealing with stress.
And maybe just to add is globally, our forest ecosystem and the tropical forests are one of the largest part of this. They're uptaking and storing a lot of carbon. So actually they are buffering about 1/3 of the total CO2 emission from fossil fuel burning and industry, which means that global change would be like 3 times more accelerated if we didn't have this big carbon sinks in the forest. Now, once the forests are under drought stress, this declines, and once we have our total balances really worked out, we can precisely nail down the point into drought when the carbon when the forest turns from a carbon source to a carbon sink. And that is very important if you look at global climate, because if we go to this edge that the forests can't really be big sinks anymore, the CO2 increase in the atmosphere will accelerate substantially.
Now, we're not separate from the forest, and in your experiments, you guys, under two different events, locked yourselves inside the rainforest and released a large pulse of enriched CO2. How do the scientists and the humans play a part in these carbon systems? And were you worried at all about your presence in the forest being affected or affecting the research?
Yeah, I think from a carbon budget standpoint, we weren't so concerned with, we do respire, we release a lot of carbon dioxide. And so we had to be careful when we were near any specific measurement points to not breathe into them and to influence the CO2 measurements we were making. But at the whole forest scale, our respiration is not critical to account for. One thing that was critical was when we're measuring volatile organic compounds in the atmosphere. These can be found at very low levels, like 1 in a billion, 1 in a trillion levels in the air, or even lower, 1 molecule per trillion of molecules of air, some very low abundances. And so we wear a lot of products like shampoo, we use shampoos, conditioners, perfume, lotion, sunscreen that smell. And so that means that they're emitting volatile compounds. And so one step we took as a scientific team was to be careful about what types of personal care products that we wore, so we weren't adding our own sort of scents and volatile mix to the system. And certainly we still did. People also release volatile organic compounds. But we did try to mitigate that a little bit to make sure that we weren't unnecessarily affecting our measurements.
And we had another question about wind. In an enclosed forest, is there wind? And how would the presence of that affect or not affect growing 30-year-old organisms?
Mm-hmm. Yeah, I can answer that. It's enclosed, and so we do have fans that circulate the air, but really our wind stress is very, very low. And that is actually translated to a reduction in the strength of some of the woody biomass that the trees use to support themselves. So some of our trees are not as structurally sound as they would be outside because they have not endured that type of wind stress. But we do have mixing with fans, and we actually have air that comes in from the outside, mixes through the ecosystem, and some of it is released. And so we're using this like an open, like a flow-through chamber, which means that we don't have to precisely control the atmospheric balance of carbon dioxide and oxygen. But one thing that we characterized was that mixing rate, because it's really important for us for measuring the whole ecosystem exchange of carbon and water. And so there's a mixing, there's circulation of the air, but there's not really strong winds in this system.
Great. And we had a question from Carolyn about the original bees and bee die-off in the biosphere. And I think that question would be best approached for our next webinar with Jane and Tabor, who were original biospherians. But in short, in that early project, we introduced a variety of pollinators into the system, whether it be ants, bees, ladybugs, and some of those species survived and others didn't. I don't have a ton of insight myself as to why the bees may have died off, but in this kind of mix of a biospheric, a biological system, and a technospheric, all the technical aspects of our facility that support these living systems, there are some opportunities for pollinators to run into trouble. And maybe speaking to that, you guys spoke about it briefly, but what is the uniqueness of having a research facility like this? And this will be our last question. Having things like air mixing and cooling and heating and rain and electricity inside a forest kind of at your disposal.
Yeah, maybe I can answer that for somebody who works a lot in many other ecosystems. It's fantastic because you are struggling so much if you want to do this type of research in natural ecosystems. You want to make a drought treatment, it rains the whole year. You want to have a controlled treatment, it gets super dry, your instruments break, you don't have electricity. I mean, field work is really very, very challenging. And you could never do something so complete like we did here in natural ecosystems. There are so many factors you can't control for, even if you try to. So I think this is absolutely unique what we have here, the opportunity we got because we could control so many things. But the one point I also really like about this tropical forest in the biosphere, that it had the chance to develop freely over many, many years. So it's not a disturbed system, it's not a plantation, it's not just some plants putting together, but the soils developed, the trees grew as they grew, there was not much interference. So it established into a natural equilibrium, which is very nice. So the processes we can look at are like in a natural forest, but we have the opportunity to control all the other factors.
Yeah, I fully agree. And I think when you consider how much control we have in this ecosystem and how large it is, how much it represents, it's a full ecosystem. There's really no other facility in the world that is like this and that is already established. That's a great point. So we're always looking to do really exceptional, answer big questions that we can use this exceptional facility to answer that just would be so difficult to tackle using other types of systems. And so I think that's what we illustrated involved in this experiment was that we can ask these big questions and answer them in a really creative and unique way at Biosphere 2. And that's what we're going to keep looking forward to do in the future with this ecosystem and this, Biosphere 2 in general, is to really tackle unique questions that we can only address at this scale here. So we were really happy to partner with Christiana and Namaya and to have so many incredible young and established scientists involved, so many students, student workers. We had student climbers, and they got involved in all different aspects of this campaign, and just incredible support from Biosphere 2 and all the staff who literally made sure we had the lights on and the water and everything we needed to do this. So yeah, we're really, really grateful to have had the opportunity and look forward to continuing to share our results.
And we'll end on that note. I just want to thank both of you, Christiane and Laura, for joining us today. And it's a real treat to be able to talk about rainforest research on Earth Day while the biosphere is closed down. And we don't have an open date soon. We don't have an opening date as of right now, but we will have one as soon as we know more about the kind of continued outlook of the coronavirus. And stay tuned. Meanwhile, I'll be jumping to our next webinar with Jane and Tabor, and you can tune into that on the Biosphere Facebook or in the Zoom webinar that is linked on the Eventbrite page. So we'll see you very soon. And again, thank you both for joining us, and happy Earth Day.
Thank you, Erin. Happy Earth Day to you.
Happy Earth Day. I can't join you on the afterwards. Here it's already evening, so.
And you have a glass
of wine. Having a glass of wine on.
Cheers.
Please enter.
In 90 years at Farmers, we thought we'd seen it all, from fires and hurricanes to animal antics we could laugh about later.
Hello. How are you? Nice to meet you. How are you? I'm Cleo. Jane Pointer and Tabor McCullum are two of the original 8 biospherians.
And after they left Biosphere, they kept this mission to help people get to and survive on Mars.
We made thousands of small biospheres about this big.
When we figured out how to make these little ecosystems stable, which was a lot of what we learned from Biosphere 2, we sent little systems to the Mir space station, and we bred the first animals, those little aquatic animals, through a complete life cycle in space.
When I went into the biosphere, I was very naive, and I thought, my experience to that point had shown me that when you put a small group of people together in a, fairly difficult environment, they pull together, and I thought that's what we were going to do. No, not so much.
There we are.
There we are, except not quite. These aren't the fancy suits, though.
They are the fancy suits.
No. These are the pre-fancy suits.
These are like the jumpsuits.
It turns out that there's a whole branch of psychology that NASA has done a lot of work in called isolated and confined environment psychology. And we were a textbook case of what not to do. One of the worst things you can do is have a team of 8. The reason is because it breaks down into factions of 4 and 4, which are extremely stable. And that's exactly what we did.
And it's really hard to describe to somebody what is that experience of living in an isolated environment. So part of the training is simply to deal with your personal baggage so it doesn't become how you interact with your other crew members, right? So that was what you start to see happening, was you start projecting, that's my sister, my brother, whatever, onto all these, and the interactions go crazy because you're carrying this stuff in your head.
Biosphere 2 wasn't the only experiment that locked people in and taught us about human behavior. There was one in Hawaii called High Seas, one in Utah called the Mars Desert Research Station, one in Russia called Mars 500. But those experiments were much smaller, and people stayed there for much shorter periods of time. I interviewed Chris McKay at NASA.
Yeah, for sure.
And he told me Biosphere 2 is one of the most ambitious projects of its kind.
Yeah.
And he said there hasn't been anything done like it since.
It's true.
Why do you think that is?
You have to think really long term before you need a biosphere.
We didn't really have problems for 6 months. But after about 6 months, you're like, I'm only a quarter of the way through this. That's just going to Mars and back fast in 2 years, right? So I'm afraid we're lulling ourselves into thinking that this isn't such a big deal when the human psychology of it and getting that right and getting that team to work right is really, really important.
Surviving on Mars is going to have to mean figuring out how to meet all of those human needs. After all, it's the hardest, longest, most ambitious trip our species has ever taken. And it turns out that NASA is actually paying attention to our psychological needs, just like they're paying attention to our physical needs. And that's because they have to. They've noticed the same psychological problems in some astronauts that the Biospherians noticed when they were inside their airtight facility. So I'm going to play you this clip. This is astronaut Henry Hartsfield describing an experience in space in a 2001 interview.
Early on when we were flying payload specialists, we had one payload specialist that became obsessed with a hatch. All I got to do is turn that handle and the hatch opens and all the air goes. It was kind of scary. Why did he keep asking about that?
So did you hear what he said?
He's just going to open the hatch. Well, he was just obsessed with the fact that one could open the hatch, right?
It's kind of like the feeling if you're standing on a subway platform and you're like, I could push this person.
Yeah.
I never have that feeling. I think about that all the time. Or being the person pushed, I think about that.
Yeah. In 2001, which is the same year as that interview, NASA and Russian NASA, which is called Roscosmos, came up with this enormous medical checklist for what to do in various crises in space. And it turns out, actually, since you have the highlighter, It turns out that psychosis is the second one on the list.
Oh, wow. Behavioral acute psychosis emergency. This is page 1 of 3. Restrain patient using gray tape around wrists, ankles, and use a bungee around the torso.
Whoa.
Yeah.
That's intense.
Yeah.
That's full, kidnapping protocol.
Full kidnapping. Administer 10 milligrams of Haldol orally.
So Haldol is a potent tranquilizer. And the thing that I find so interesting about this isn't exactly what you do, but the fact that they find this so important in the first place. And that really surprised me. I went to Utah and I learned about how toxins in the soil are something we're gonna need to figure out if we want to survive on Mars.
I talked to Joss, and we talked about how your blood is going to boil if you stand on the surface of Mars. And now I find out that actually the thing that might be most dangerous to us is just ourselves on a mission like this. But at the same time, even though I learned all of that, I also learned that there are possible solutions to every single one. And I think that's a pretty good reason to try.
Humans will go to Mars. Humans will explore Mars. That much we already know. Whether we will stay there on the long term is a question that we have to answer by trying.
And by trying to go to Mars, we could have a role to play in a mission for survival that's much, much bigger than just us.
If you look at the universe, the thing that looks like could be the basis of value and goodness in the universe is life. It's the most amazing phenomenon we know. We're the only species within that domain of life that can comprehend the concept of planets in space. So maybe we have a role to play.
We all eventually do want
have
humans to get
maybe
to Mars.
some of the
I
most
see everything
intimate
that we're doing
history
now
with
is
this
preparing
facility
for
out
humans
of
to get
anybody.
there.
That's almost the essence of life.
And
is to
it's
spread
such
to
a
new
treat, and I'm
habitats.
going
So
to
it
introduce
seems like
them in
we're
just a
doing
second
what we're
once
supposed
we
to do.
let
And
the
when it
attendees
comes to
come
Mars,
in,
at least as
once
far
we
as
hit
we
that
know, we're the
12
only
o'clock
species
mark.
that can.
But as you get situated here on the Zoom webinar,
Yeah. Oh no. Let's go. Go, 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 go. Okay. All right. Let's get.
Yeah!
We're gonna probably.
We were not meant to survive out here, but we did. We did it.
Silicon carbide from Wolfspeed will allow innovators to go farther.
and I'm, really pleased that we all get the opportunity to ask these two questions. And beyond their role as crew members and designers of Biosphere 2, they've led a very rich career in kind of commercial spaceflight and space entrepreneurship. So Jane Pointer is a space entrepreneur, Biosphere 2 crew member, author, and speaker. She was a co-founder and founding CEO of Paragon Space Development. She was also a co-founder and founding CEO of Worldview, and is currently a founder and co-CEO of Space Perspective. And Tabor, also a founder and co-founding CEO of Paragon Space Development, Worldview, and Space Perspective. So you guys have been partners in crime for quite a while, both as crew members at the Biosphere and co-founders on a variety of business ventures, particularly revolving around space. And just briefly, these businesses, Paragon and Worldview, they're involved with sending hardware to places like the International Space Station, deep dives in our world's oceans, as well as kind of some of the first commercial attempts at colonizing the Earth's stratosphere. And hopefully we'll be able to get some more modern context to some of their modern ventures. But I think there's a lot of interest today with Jane and Tabor's early origins at Biosphere 2. So we'll start with that. And I guess my first question, starting off in the honor of Earth Day, is what is a biosphere?
Well, that's actually an interesting question, because we had to wrestle with that at the Biosphere 2 project. It's sort of hard to remember, and some of you actually don't even remember, that the word biosphere was not in the popular lexicon, right? That idea that the Earth has a biosphere around it, that's sort of a semi-self-regulating living system, was a pretty new idea that Biosphere 2 was actually a part of really introducing. So a biosphere, as we defined it, is maturely closed. So the Earth's biosphere is maturely closed by gravity. Biosphere 2 is maturely closed by glass and steel. energetically open, so
Clean,
radiant
increasing
energy
out-
comes in to grow the plants and provide energy to the system, and informationally open, so we can send information in and out. And so that's the fundamental definition of a biosphere.
So what we thought we'd do, Erin, is go through a few slides, show some baby pictures of Biosphere 2, talk a few minutes about what it was like to be inside and isolation, and a little bit about what we're doing, and then open it up for questions, if that works.
That sounds great. Yeah.
Good. Hey, everyone.
Hello. So the image that you're looking at now is one of the first concept drawings we did of Biosphere 2. Of course, this was before the days of computer-aided design. And so the idea was to test this hypothesis that an artificial biosphere could be made from the ecosystems that comprise the Earth's biosphere, and that resulting biosphere could be both a laboratory for the study of ecosystems, as well as a way to colonize other planets, take life to other places in the solar system. So Biosphere 2 in that theme had natural ecosystems from the tropics, rainforest, ocean, savanna, thorn scrub, marsh, and desert. And of course, because it had people inside it, we needed an agriculture and a habitat. And it was designed to all be airtight, sealed, which we got to about 8% gas exchange per year, which is actually pretty extraordinarily tightly sealed. And these things called lungs allow the air to expand and contract with temperature inside.
So it was a super exciting idea that we were attempting to create humanity's first other biosphere. Of course, Biosphere 1 is planet Earth, Biosphere 2, the structure that we all know and love. And so here is an early couple of baby pictures of Biosphere 2. As Erin mentioned, Tabor and I were both on the design team. Tabor was involved and in charge of the analytical systems, the analytical chemistry systems. I was in charge of initially of the insects, and then took over the agricultural system. So it was sealed on the bottom. I'm sure many of you have toured it. It was sealed on the bottom, still is today, with steel all the way under. You don't see it all the way through because that's then covered in concrete. And then, of course, sealed above ground with the steel and glass. And one of the biggest questions is, how are we going to get this thing sealed as tightly as it needed to be in order to be able to be a hermetically sealed biosphere? So I will remember the first time I saw Tabor at the Biosphere, where they were they were working with one of the very early seeming approaches, which was butyl rubber, something like that. It was like this really black, gooey substance that they were pushing into all the seams. Now remember that glass and steel obviously have different thermal expansion and contractions. And so as the biosphere heats and cools during the day, the whole structure moves by several inches in the summer. And so they were having to deal with that movement, which is why they started out with all of this black gooey stuff that was all over everybody trying to install it. Clearly it didn't work. So they ended up with this really interesting technique of having the panels of glass essentially floating in sandwiches of silicon. And I think there's an image coming up of sealing all of that in a minute.
So Biosphere 2 sort of had to be constructed sort of an instant biosphere. So we collected plants from Venezuela, from botanical gardens in England and in the US, and from around the world, and planted and assembled the biosphere along with then all the big panels and then the glass that went on top of that. So it was like a huge tinker toy project.
And that, of course, is putting a tree into the rainforest.
So on the left, we're sealing those floating frames of glass. There is 60 miles of glass edge seam in Biosphere 2. And all of those seams are maintained and tested with the vacuum chambers. It was a huge effort to seal up the biosphere.
And then on the right is putting the very first life into Biosphere 2, which was the soil that went into the farm.
The marsh there is actually simulates about 150 miles of estuarine system from freshwater to saltwater, and each of these divisions that you see on the left-hand side there is a different salinity and ecosystem that all then works together with a water flow system. So on the right there, you're looking across the savanna, can't really see the ocean at the beach at the far end, and the rainforest is off in the distance.
So the marsh was collected as cubic meters of extant living ecosystem of a marsh from an area in Florida that was actually being turned into a parking lot. And then Tabor and I just really had the harsh duty of collecting corals, which really meant that we went to the Bahamas and the Yucatan to collect corals. It was very odd experience because when you collect corals, you're effectively down on a coral reef with a hammer and a chisel collecting whole heads of coral that we're then bringing up and putting into the ocean. We were collecting these corals, though, from areas that had already been badly damaged by hurricanes. And we also had to go and collect all the fish. So I was also in charge of collecting fish that went into the biosphere, which was an interesting ordeal.
So then after several years, we started the project conceptually in sort of 82, 83. Buckminster Fuller essentially double dog dared the team to build a biosphere and then began construction. Groundbreaking was 87 and we went in 91. And so this is shot shortly after completion.
And there we are before we went in. And then here we are going in. Yeah, it was a big day. It was actually very funny being, right up to the moment as we were walking in that airlock door that you can see us walking into there on the left. There were literally 100 people running out the back airlock, out the back of the biosphere. So the moment that we walked in was the very first moment that the 8 of us were inside Biosphere 2 alone with no other people.
And now you're starting to get to what that feels like.
So we thought we'd show you a little bit about the farm. If you've been to Biosphere 2, you're very familiar with all the different ecosystems, but not the agricultural system, which is not there anymore. It's got a great experiment inside that I won't go into now. So here's just a few shots inside the farm, or the intensive agriculture, as we called it. So we are collecting at the top left there peanuts. And one of the really sort of cool things when you think about holistically about a farm like this, when you really try and eke every calorie out of it, and also think that we had to not just get every calorie, but make sure we had enough fat and protein and all the right amino acids and everything. And so we took all of the food that we didn't call food. So in this case, it would be all the greens from the peanuts. So on the top right there, we're actually taking the peanuts off the peanut plant. And then on the bottom left, I'm feeding them to the goats, which of course would give us amazing milk. And the peanut leaves turn out to have very high protein for goats. So the goats were amazing. We also had pigs and chickens. And then on the bottom right is our doctor, Roy Walford, who is threshing wheat as well.
So as everybody know, one of the big questions we had going in was, how does the chemistry of a biosphere work? So my job was to try to figure out and understand the chemical cycles going on, especially in the atmosphere. And you may have heard that we lost oxygen in a way that we didn't understand. We originally thought that we would every time we lost oxygen, we'd gain carbon dioxide, and carbon dioxide would be the real problem. But we were losing oxygen without an appreciable gain in carbon dioxide. And I can go into the chemistry of that later if somebody wants, but in essence, it was an interaction we didn't appreciate before closure between the soil and the concrete structure inside. And so I am on the left there taking gas samples from the soil to be analyzed back in the lab. And on the right there in a laboratory that we designed to be inside Biosphere 2 to do all the chemical analyses that we need to do on the air and water and soil and plant tissue and things in the biosphere.
Another area that actually you won't see at the moment walking through the biosphere is sort of what we call the lowland savanna. We actually call it also our biovalve. So on the left-hand side there, you'll see 3 biosphereans standing in the midst of the grass of that area in full growth. So it's pumping oxygen into the atmosphere, it's scrubbing CO2 out of the atmosphere. And then on the right there, we have stopped raining on it and thereby effectively turning off the biovalve. And now it's having almost no net effect on the atmosphere inside Biosphere 2. We didn't have any animals to go along and harvest it all. So we were the herds of animals harvesting it. But actually, rather than turning it into carbon back into the atmosphere, we banked all that carbon. Because our CO2 was a bit too high in the biosphere, we actually took that, all of those leaves there, and stored them in the basement. So we were practicing carbon sequestration inside Biosphere 2 way back then.
So this is, of course, one of our favorite topics in the biosphere, which was eating. We didn't always have as much food as we wanted, and we all lost quite a bit of weight. The room on the left is where we let bananas ripen, and that room was locked, lest we go in at night and steal the bananas. We also found it really important socially and for us to have a feast sort of once a month. And so we had 8 birthdays. That was pretty easy. We were pretty well distributed, and a few holidays, and we had to make up a couple. But this is one of those times where we all got together and had saved food over the course of that month to make a big feast. And there's sweet potato pie and breads and peanuts and rice and vegetables. And I think one of the pigs is there too.
So this gets us to talking about being in isolation. So, which is a fairly topical issue of today. We were somewhat prepared for being in isolation. We were somewhat trained for being in isolation, and we were there for 2 years and 20 minutes. So let us hope that our current situation of us all being thrust into isolation is a lot shorter than that. So it turns out that there's a whole branch of psychology that NASA has studied called isolated confined environment psychology. And one of the things that happens in the Antarctic, where they have plenty of food, is food hoarding. And of course, we had food hoarding and people food stealing. Thus, the bananas were locked away. It was the only room in the entire biosphere that was locked, actually. And seminal experiences. So one of the things that one of the reasons that we had these monthly feasts was because they create seminal experiences, things that we can all come together as a group to do that's completely different from everything else and sort of breaks up to a degree, monotony of being in isolation.
This was similarly a party on the beach. We actually brought a TV down once and put one of those little videos of a fire, and we would catch ourselves warming our hands on the video. But again, these were sort of the events that were important to us to break up what was a pretty monotonous, hardworking day-to-day routine.
So one of the things about being in isolation is, well, particularly for us inside Biosphere 2, when it was social distancing. I think now we're calling it social distancing, but actually I think what many people are finding, it's more physical distancing than social distancing, as long as we're reaching out into our communities and staying connected in the way that we are today. But regardless, it can get monotonous and to do it to a degree somewhat boring. And what can come out of that is actually creativity, a tremendous amount of creativity. And we planned for some of that. So Tabor and I took in musical instruments. That's us on the right. It really doesn't matter how good or bad our art was while we were in there. The point was we were doing it, and it was great. We also loved to play with this idea that we were in Biosphere 2 and the rest of humanity was in Biosphere 1. And so we had inter-biospheric art festivals, us doing our amazing art on the inside and exchanging that with artists on the outside. Sometimes we jammed down the phone. Remember, there wasn't the internet really in the way that we experience it today at all. When we were in there. So we were jamming down the phone. So there's a lot of tools now that we all get to use, like Zoom, that we just didn't have then. And then on the left, Tabor, do you want to explain the one on the left?
Go ahead.
So Roy Walford was the doctor inside, who I mentioned earlier. And he was a spectacularly well-known gerontologist. And has done huge amounts of work. You may be familiar with the high-low diet, the high-calorie, the low-calorie, high-density diet, which, as it turned out, we ended up being on, which is a whole other story. So his goal when he went into the biosphere was to go in as a scientist and come out as an artist. And so he was constantly coming up with all of these crazy things for us to do. And one of them was this on the left-hand side here, which you will not see. It was taken off the wall. It was done with body paints. And those are full-sized imprints of people's backsides and their legs on the wall, making the butt-wheeled wagon that Tabor and I are the two characters in the front, outlined in green, and so the blue are the wheels, and then the red are the joined hands of the men and women that were the wagon that was, going to go to the stars. So he had this whole story around this thing, which was really
fun. This is several of us taking a bit of time off, and the crew did split into a couple of sort of factions that preferred to hang out with each other. I guess is the easiest way to say it. Certainly, the psychological aspects were indeed some of the hardest parts of the Biosphere 2 experience, but we're having a good time here. And then after 2 years, we came out. The answer to the question one of the questions that we built Biosphere 2 to test was, indeed, it's possible to build an artificial biosphere, and it works. Of course, we've all heard about some of the technical issues that we had in the biosphere, like we didn't quite have enough food, and where did all the oxygen go? Well, it turns out that it's a very hard hypothesis to test. You have to have built the biosphere well enough to know that you can build a biosphere that works. And what we found in Biosphere 2 was that all the issues we had were part of the design of this biosphere, not intrinsic to the act of building a biosphere. So today we have Biosphere 2 as a research lab, in no small measure because we found that we can make these small ecosystems in a closed system that's airtight, and they thrive and work and predict and work in ways that one would understand.
One of the things that we then did is we wanted to actually prove to ourselves that you can, in fact, make small, tiny, in this case, tiny biospheres, and have them persist for extended periods of time. Biosphere 2 is an N of 1. And it's really hard to prove anything definitively with that. And so we created hundreds, thousands of these little tiny, on the top right there is a small ecosystem that we created and sent. This is actually being unpacked on orbit. I think this is going into the SPESA module on the International Space Station, where the animals inside there were the very first time that any animals had gone through multiple generations in space, and we had that ecosystem alive there for 18 months on the International Space Station. The picture below is us messing around with developing those systems. I think we're pressure testing one, it looks like, before it goes. Oh, that's actually one of the systems before it goes up. And in fact, we've had one of these just anecdotally at home that we made ourselves closed for 20 years now, and there are still breeding populations of aquatic animals inside, which is pretty cool.
Paragon here is a company that we started with some of the people after we came out of the biosphere, and it does a variety of technology developments like life support systems. It has the parts of the life support system on Boeing's Starliner. It has a water recycling system going to space station this year, and a variety of technologies used in spacecraft. And so it really is part of the legacy of Biosphere 2 in the work that Paragon does.
And one of the things that we did was to break the Red Bull Stratos jump. Some of you, I'm sure, have seen Felix Baumgartner stepping out of the capsule and spinning wildly as he falls down to Earth. We actually work with Google executive Alan Eustace to take him up to break the jump. You wouldn't necessarily have heard of it because he wanted it, very on the down low, being a Google executive and having a family and all of that. So we did just that. 2 years after, in 2014, 2 years after Felix's record was set, we set that record. And so on the top left, you can see that's the actual balloon that we took him in, and you can see just at the very tippy bottom of that hole, that's all balloon, by the way, that you're seeing there. You're just only seeing the lift gas at the very top. It expands to fill the entire balloon, which would be almost the size of a football stadium once it's fully expanded at altitude. We took him up to 136,000 feet. The bottom left is him at altitude, having just been released from the balloon. So you can see the sky is, what?
Bottom right.
Oh, I'm sorry, bottom right, other left. So you can see the sky is completely black, and above him, that's him in the spacesuit, actually just as he was launching, calmly waving at us all on the ground. And then the bottom left was actually during a test jump out of an airplane, and that is the only time you will ever see rollerblades on the front of a spacesuit. This is a full spacesuit that our team designed. And the reason he had rollerblades on the front was that the exit of the airplane was too short for him to stand up and get out of. So he literally had to lay on his front and be pushed out the plane. Thus, rollerblades on his chest.
We took a lot of that work and started a company called Worldview that was originally going to do sort of a space tourism, taking people to the edge of space. But in the process of doing the Stratix and other work, we found some really interesting layers of atmosphere that allow for navigation and station keeping. And so Worldview concentrates on providing a platform for communications and remote sensing. The picture on the left is getting ready to launch from our facility just south of the airport in Tucson. And on the picture on the right was actually taken by a telescope at University of Arizona. This is that vehicle at about 75,000 feet with buoyancy control and the telescopes and the like at the very bottom.
And last but not least, we're now working on space perspective that we can't really talk a lot about yet, but it is a human spaceflight company. Very pertinent, I think, for Earth Day, because we really want to take a lot of people up to space so they can really experience our planet from that vantage point, to really get that planetary perspective that we all need to really address and appreciate our planetary challenges today.
And one of the parts of the experience that we had in Biosphere 2 was to sort of get what astronauts talk about. So when you talk with an astronaut about their experience in space, they talk a lot about seeing the overview, the world as a system, And in Biosphere 2, we essentially had view of an understanding of the system that we were in sort of from the inside in our small biosphere. And so we think it's important for people to have that experience more generally and accessibly to see really how small our world is. And so that's what this is for. So Aaron, that's it for our kickoff here.
Great. Thank you so much. Some of those pictures, I'm sure many of us haven't ever seen before. So it's a real treat. And speaking to what you were just talking about, my personal question for you guys, when you're in a biosphere, in a closed system, and you feel the effects of losing oxygen and your breath is connected with your environment, how does an experience like that, for as long as you guys did it, how does that change your perspective walking outside and just day to day? And do you still carry that kind of intimate connection with our environment now?
It's clearly harder now. We certainly carry it now. In the biosphere, it was so evident how tightly coupled it was. We could make a mistake with the way we handled the agriculture or composting system or something like that, and you would see the results in the atmosphere that afternoon. Or once there was an eclipse, and we could see for the time of the eclipse how the biosphere stopped photosynthesizing. We turned orange because of the sweet potatoes that we ate, and so we were exchanging carbon with the biosphere. And certainly, we carry that with us, but it is certainly harder in a day-to-day sense now because the Earth is sort of so seemingly big.
It's interesting, right? So we did have that incredible visceral experience that I think some of us are experiencing now, right? It's very clear how interconnected our planet is. And if people are plugged in, they're also seeing the incredible effect that humanity staying at home is having on the planet. We're actually having the eclipse effect right now, right? We're seeing all kinds of things. Pollution in water is cleaning up. The skies are cleaning up. Lots of things that we're beginning to see because humanity is such a huge force on our planet today. So I do think we're having that moment now. And what's going to be interesting to see is how that carries forward. So there's actually been some interesting work done looking at astronauts, and I'm sure some of you have heard of the overview effect that Frank White sort of developed this concept, which really is, in its simplest form, you go to space, you get this incredible experience of seeing the Earth in space, and you're suddenly smacked in the face with sort of how small our planet is, how interconnected we are. But what's interesting is that there is a measurable effect. There is a significant effect, and that astronauts who return from space actually tend to get more involved in social and environmental causes afterwards. And it's a statistically significant increase. So what's going to be interesting is to see if there is any sort of lasting impact from all of this tragedy that we're experiencing on a global scale right now. That's going to be interesting.
We have a question from Ronald, and he asks, what would you want to see in a Biosphere 3 project? And what is the future of synthetic biospheres in space? So kind of a modern take on Biosphere 2.
Well, so it's sort of interesting, all life as we know it is in the context of a biosphere, a system of living organisms that are interconnected energetically and materially. So a Biosphere 3 would take all the lessons that we learned in Biosphere 2 and in ecosystem science and get closer to, call it the optimal design. We had imbalances in the way we stored and handled carbon and things like that in Biosphere 2. So I think there's a lot to learn there. There's also much more computing power now, and we can do a lot more with understanding how the system works on a modeling effort. That, you know, we didn't have the computing power then. You know, this little thing called the 286 came out with with floppy drives just before we went in the biosphere. That's what we ran Biosphere 2 on. And so both the current computing power, AI, and helping understand how the biosphere is working, and then in space biospheres, and the company actually that built Biosphere 2 was originally called Space Biospheres Ventures. And we were building about the same time that the International Space Station was being built. It's sort of a fun way to think about biospheres. You can take all the genetics of a biosphere in a very, very small container and build a biosphere somewhere from all those seeds and frozen embryos and all kinds of ways to transport the information that then builds a biosphere in space. So I think we'll find ultimately that that's how life expands through the solar system and beyond, is in the basic unit of a biosphere.
And a question from Robert Christofferson. He asked, did each of you have a special place, whether it be a hiding place or just a place to gather thoughts within the biosphere when you were enclosed?
Sure. Yes, I loved sitting on the beach at night. Like at sunset was my favorite time, or at night when it was starry out. Because when you sit on the beach and you can hear, you know, the sound of the wave machine, if you've been in there, it's this really strange sound. And then at sunset, you look out the windows and you look out through the space frame, and the space frame and the whole shape of the biosphere starts to feel a little bit like a Gerard O'Neill space castle. So if you look out the windows, you could for all the world suddenly be on Mars because the Catalinas turn red. And then at night, when you look out and you see the stars, you could be perhaps on your way to Mars. It was just a really wonderful way to have a sort of a flight of fancy, sitting on this human-made beach in Arizona inside this incredible biosphere.
We didn't describe the habitat much, so the question brings that up too. We each had our own little apartments, sort of a sitting room and bedroom. There was one kitchen that Erin is actually sitting in now, the dining room. We had a medical facility to monitor everyone's health and to take care of any issues that brought up. An analytical laboratory, a machine shop, sort of an office, mission control space, and gathering spaces. So the habitat area of the biosphere is actually pretty complex in and of itself.
And we have a question from France, and they're thanking you guys so much for being a part of this today. And their question is, was there a wind generator inside Biosphere 2? And perhaps you could speak more of all of the technospheric support for these living systems that created the biomes in that answer.
Sure. Wind is actually very difficult to make because it takes so much energy to do it. But we had air handlers that serviced the air by filtering out particles, taking humidity out of the air to make water that would become rain. And if you took all of those air handlers together, they moved about, sorry for the English units, 6.5 million cubic feet a minute of air, which is roughly the volume of Biosphere 2 every minute went through one of these air handlers. And even with all of that 6 million cubic feet of air a minute moving through Biosphere 2, when you were in the biosphere, for the most part, it was sort of just a wispy breeze. And one of the problems that a lot of the plants had in the biosphere was there wasn't the wind that would move the tree limbs back and forth and create stress wood. So they got these sort of tall, lanky trees that sort of wanted to fall over. We wished we had a lot more wind, but it was very difficult to make that much wind. You don't really appreciate the energetics of wind until you have to actually make it.
And then if I can perhaps speak further to the energy production. So the concept early on was that we were going to produce the energy for the biosphere from solar. Being in Arizona, that seemed like a bright idea, except at the time it would have taken acres and acres. Remember, we were designing Biosphere 2 in the 80s and building it then. It would have taken acres of panels and would have cost an arm and a leg or so, or 3 or 4. And so we didn't do that. And so we used grid power, and we actually had a backup energy system outside in the event the energy went off. With sort of from the idea, sort of philosophically, being that, we're thinking of a biosphere being energetically open. And so we were getting the energy for our biosphere from outside.
To answer the technosphere question, we called it the technosphere, and somebody said Biosphere 2 was like Eden on top of an aircraft carrier. In the basements underneath pretty much the entire biosphere are a wide variety of technical systems, monitoring systems for the temperature and humidity and air composition, and systems to heat and cool the air, and water processing systems, and water recycling. So there's a lot of engineering that goes on to keep a biosphere running when you don't have sort of the resources of the whole Earth helping balance the temperature, for example.
Another question I have for you guys, during the time of the experiments, a popular term was the concept of cybernetics, which is kind of getting this technological feedback in situ or in real time, and letting that allow the steering or the guidance of the biosphere as a whole. How much of that was really going on during the biosphere in terms of changing parameters of your biomes for certain production? And on some level, were you guys the cybernetic systems just observationally in the changes that you would make as farmers and stewards of these landscapes?
Yeah, so both is true. We did monitor as far as was practical at that time. Every aspect of the biosphere, the gases that were in the atmosphere, number of gases that were in the atmosphere, and the humidity, the temperature, things like that. Of course, we knew the levels of water in different tanks and ponds and the ocean and all of that. So there was a certain amount of automation, but not a lot, that was providing feedback, just because all of that kind of thing was so early. I mean, remember when Tabor was designing this is crazy to think about. When Tabor was designing the analytical chemical lab inside Biosphere 2, his was the very first laboratory that didn't use paper printouts from machines. You literally had to write code so that he could not have paper, because we couldn't have any paper in the biosphere. So we didn't have those kinds of capabilities of all of these sophisticated algorithms and things like that we do today. So yeah, to a large degree, we were the cybernetic feedback. And we were the ones going, okay, we're not going to water over here today because we don't want to have a spike of CO2, that kind of thing.
Playing into that, we had a question that kind of wanted to focus on the social isolation. And it's so relevant for people logging in now. And you two spoke a little bit about how creativity was an outlet for you in isolation. But what are some tips for us as quarantiners here today as we experience our own social isolation?
Sure. So I'll start off with that one. So being inside Biosphere 2 was a little bit like sort of, I like to call it a monastic experience for somebody who really didn't want to be in a monastery. And we thought we had really practiced that and understood it by spending a long time on ships and back in Australia. But it was really different when we were sort of confined the way we were. And one way to sort of think about that seemed to fit for the way my experience was that it was like I carried in a whole bunch of emotional baggage, and sort of the routine day-to-day sort of lack of distraction, lack of movies to go to and things like that, sort of brought those things to the surface, things that I didn't really even realize I was sort of carrying around. And then that would create, flashbacks, with these very vivid flashbacks, which is, I'm sort of back in a scene that happened when I was younger, but it just seems very vivid, more than just a memory. And people in Antarctica wintering over report that. And sort of obsessive things like, I couldn't stop thinking about this high school sweetheart. And it was, we were sophomores or something and broke up in a way that wasn't very, was, I guess, typical for a teenager.
Very mature, I'm sure.
Yes, absolutely. And I couldn't stop thinking about her. And I mean, and it was sort of like getting to the point where it was getting ridiculous. I mean, like, this was 10 years ago or something. And so I finally tracked her down and got on the phone and said, hi, yes, it's me. Yeah, inside, you remember me from, yes, Biosphere 2, this whole hilarious scene ensued down the phone from inside Biosphere 2. Well, I just had to say I was sorry. And then it all went away. But it was like this piece of baggage I had to let go of. We had things where, I would be mad at somebody for reasons that I didn't understand, that was sort of connected with some piece of social, emotional baggage. So I guess the advice is, as these things happen, you sort of have to deal with them in real time. You can't really just try to sweep them under the rug, because in my experience, it just sort of got worse and worse. I would obsess more and more, or this thing would bother me more and more. And, we finally called up psychiatrists from inside the biosphere and, I had to interview them and say, are we going crazy? And, be on the phone with, psychologists saying, yes, it's me in Biosphere 2, I need help. We weren't. These are all pretty natural reactions to being isolated and confined in Biosphere 2. But it helped a lot. So, reaching out to people, reaching out to friends, getting people to help you make objective assessments about what's going on. Those are all pretty important.
So I think in sort of some of the takeaways from what Teva was just talking about is that it is really hard being confined. There's no two ways about it. We're really lucky here because we get to go outside. We're right next to the beach. We get to walk up and down the beach. And so that we're really lucky. But even then, we're confined here. And even now, it feels a little bit stifling, right? So I think the first thing that we all have to recognize is this is not normal. And it's okay for it to feel really not normal. And that in and of itself is kind of a relief. I mean, I remember when we really, that the time when I finally said, okay, that's it. I need help. I need professional help, was when I was one of those flashbacks. I'm standing in the farm. I'm standing in the sweet potato field. And all of a sudden, boom, I'm back to when I'm 8 and I'm having this terrible fight with my brother. And it wasn't just like, daydreaming. It was like it was happening. And then I'm back and I'm like, oh, where did I just go? This is really strange. I think I just lost my mind. So, we do find ourselves professional help. And this woman says, look, if you weren't having a strong reaction to the situation you're in, I might actually think you had something wrong with you. So I do think it's really important to recognize that this isn't normal and we're going to have difficult reactions to it. So that's the first thing. And as Peter said, reaching out, it seems, I wish I had bought Zoom stock because it seems that instinctively now we have the tools and we are instinctively reaching out to people. Like I've found that I, particularly in the first couple of weeks of isolation, was reaching out to people I hadn't, spoken to in a long time and vice versa. And it turns out that is incredibly healthy. We remember we are physically isolated, not socially isolated. So all of those things are incredibly important. First thing to remember, this is not normal.
They did an analysis, as is topical, also kind of a bit more, looking at what they call behavior settings. And so behavior setting is like the office, you have people that are different and a subject, a thing that you're doing that's different. The gym is a different behavior setting, home is a different behavior setting, going out on vacation is a different behavior setting. Biosphere 2 was just one, and most people have several. And it was sort of characterized by the doctor we worked with afterwards as behavior setting deprivation. But we sort of had 1.5, and that half was electronic communication with people outside. Email, we did have this funny little thing. It was a bit like a fax machine on a screen that very slowly sent an image over. It took about 4 or 5 minutes to get one black and white image. But yeah, having people to talk with outside, friends that I could joke with, making that outreach was really important, and in the behavior setting side, sort of created stimulus and input that was new and different and very helpful.
And we're closing in on maybe our last 10 minutes or so here. And we have a question from Bill Stubler, who asked, was there anything about the biosphere experience that was much easier than expected once you got inside?
That's a really good question.
Apparently not, because neither of us can think of anything. No, the whole thing was...
Well, I can remember.
Oh, you can think of something.
So, but it only became apparent when we came out. And we when we came out, we called it social spaghetti. Inside the biosphere, it was pretty straightforward, right? There was mission control, there was the people we knew outside we could call and each other, and we all sort of aligned along sort of fault lines of how we thought we should run Biosphere 2, and that was sort of the crux of a lot of the conflict in the biosphere. But it was all very sort of, you knew who the people were, it was all pretty straightforward in where those fault lines were. We came outside, I mean, society is just phenomenally complex. And there's all the different people that talk to different people that say, I mean, it was just sort of overwhelming. And then we also realized how useful habits are. But we didn't really realize that till we came out. There's the habit that puts the keys back in your pocket when you leave the car, or the habit that puts the wallet back in your pocket when you leave the restaurant. All of these habits went away. And so we were just like completely helpless at points outside when we came out, because we didn't have all of these handy little habits that sort of keep the processes of everyday life going. So in those aspects, life was sort of simpler, and yet we didn't have trash, as an example. We spent 2 years without trash, and we came out, we threw a party for a bunch of friends, and we were astounded at the amount of trash that one party can generate. We were also astounded at the variety of food. I mean, it would take us 3 months to make a pizza. We had to make the cheese, we had to make the dough, we had to grow the wheat, all of that. And you go to a grocery store, and there's there's cheeses from across the world, and there's wines from Argentina and Portugal, and it just goes on and on and on. And the amount of and variety of foods that we have available to us, for the most part, in society is just astounding when you go from sort of the simplicity of growing it all yourself.
It certainly reminded us how incredibly abundant this world is. For most.
We've gotten a couple of last significant questions about toilet paper, and you mentioned that there was no paper available. So how did that work in the bathroom?
Actually, I liked it a lot. We had sort of a little portable bidet. It was like a little thing with water that came out of it. So it was I rather liked it.
Yeah, you just wash with water.
Yeah, you just wash with water.
It's really much easier.
And we've got a question from Joaquin Ruiz, who is the VP of Global Affairs and the Executive Director of the Biosphere. He asked, how would you create the perfect team to be in an enclosed system like Biosphere 2?
Oh, that's an interesting question. Yeah, so I think there's 3 different things to think about. So obviously, selection is really important. Russia got pretty good at selecting, has gotten pretty good at selecting people to go to space. NASA has certainly gotten pretty good at selecting selection criteria for who to do these kinds of things, particularly the long duration ones. They're getting better at. Training is really important, and not just physical training, it's emotional and mental training. We were completely blindsided by how difficult those 2 years were going to be emotionally. And then the third thing is giving them the right support. Interestingly, astronauts have discovered that mission control can be quite a meddling entity, and actually be not supportive or very supportive, and it can have an outsized impact on the team, how mission control behaves towards the team. You become hypersensitive in an environment when you're enclosed for a long period of time, it seems. And things tend to get a little out of perspective. So that's incredibly important, is your support structure while you're in as well. So it's not only the selection. It has to be 3 things. It's the selection, it's the training, and it's the support. Because if those 3 things don't all work together, it will become quite tricky.
Yeah. As Jane said, the rest have become pretty good at that selection part, but I tend to think that part of the training aspect that Jane talked about is actually putting people in isolation and see how they do. Because it's really hard to understand it unless you've done it. And you wouldn't want sort of the first time somebody's experienced long duration isolation to be, on their way to Mars. And so, we tend to get these very type A people, oh, I can take care of anything, I can handle anything. And we were sort of that way going into the biosphere. And then with the crew after us, we said, no, you need to, do these things, have this psychiatrist that you work with. They gave them all this advice. And the response to our advice was uniformly, you guys are just wimps. We're the type A ones. We'll do great. And, a few months later, they were calling us up with all the same problems we had. So part of it is just, training has to include experience.
You guys, thank you so much for joining us today. We've got to run off to our next webinar with a couple of modern coral reef biologists. But I think I would join others in saying that it's just such a pleasure to be able to talk to you guys on Earth Day and just hear firsthand about your experiences in the biosphere and how they're so relevant to what we're experiencing now. So thank you so much. And I would encourage all of our attendees to look up Jane and Tabor on social media and look into Worldview and the space perspective as well. And we'll see you on the next webinar here starting very shortly. So thanks again, guys.
Thank you very much.
Thanks, everyone. Bye.
open up our Q&A box and our chat box. And if you're joining us, this is our third webinar of the day. Thank you for participating in Biosphere 2's Virtual Earth Day. We've had talks from rainforest ecologists, from original biospherians, and now our finale webinar is with two great scientists that are working on a project in the ocean biome, and they're a part of the Biosphere 2 Reef Lab. And I'll just wait a few more minutes before I do introductions. But I want to encourage everybody to right away start asking your questions on the Q&A box and in the webinar chat. We love to hear where you're listening from. We've had folks today from France, Norway, Brooklyn, Tucson. So it's really fun to hear from you guys and where you are around the world. So without further ado, I want to introduce Dr. Diane Thompson and Katie Morgan. Diane, excuse me, 1 second here. Katie Morgan, why don't we start with you? Katie is a manager of marine systems at Biosphere 2, and she got her degree from the University of Arizona. Diane, I apologize, I'm having a hard time finding your description, so I may have to ask you to talk about your background briefly at the start. But Katie's been a marine manager at Biosphere 2's Ocean for several years, and she's kind of the on-the-ground presence for this reef system. Has been involved in the planning process for the Biosphere 2 Ocean since the modern campaign to really start to bring this project into a context of creating a resilient reef research center here at Biosphere 2. And you can see in her viewer window that she's actually got the B2 Ocean in her background. So she's coming to you live from the beach of Biosphere 2. And Diane, again, I apologize. I'm having a hard time finding your description, but Diane is a professor at the School of, is it Natural Resources, Diane?
Geoscience.
Geosciences.
Science. Okay.
I'm gonna let you talk about your academic background briefly yourself.
Clearly, I just can't be introduced. So I, as Aaron alluded to, I am an assistant professor in the Department of Geoscience, which is in the College of Science at the University of Arizona. And I am the director of marine research at the Biosphere 2. So I work closely with Katie and our team to transform the Biosphere 2 Ocean into the state-of-the-art reef lab to really try to develop solutions for coral reefs under a changing climate. And as I'm sure we'll get to today, my own research program focuses on using corals to really better understand how oceans have changed in the past so that we can better predict how oceans will continue to change and in turn help buy some corals some time to ensure that we have reefs in the future.
Great. And I guess what I'd like to start with in honor of Earth Day with a very simple question: what is a coral?
It's a great question, Aaron. It's often the first question that we get, in part because it's a bit confusing, a little bit misleading. So in a lot of ways, corals, although they are animals, have a lot of rock-like and plant-like features. So here's a picture of what a coral organism looks like, a couple of individual, what we call polyps, the individual coral organisms. So these are really closely related to jellyfish and sea anemones, or anemones if you're Nemo. And so they are animals, and they do feed with these tentacles that you can see here. So they have stinging cells that actually capture prey and bring them into its mouth. But actually, a lot like plants, they also have these algae that live in their tissue, and that's what's shown here. And these small, what we call symbiotic algae, so a symbiosis means a relationship that's mutually beneficial. These algae provide actually the majority of the coral's nutrients, their energy. And so these are really critical for the health of the coral organism. And in turn, the corals provide a safe home and waste products that actually help with their ability to produce energy. And then finally, what we know of coral reefs is they actually have these great rock-like features. So just like the shells that you find on the beach, corals produce a skeleton that's very similar to that same mineral that you find on the beach. And produce these massive structures that are so critical for providing habitat for a diversity of organisms, and ultimately even protect our coastlines from storms and growingly sea level rise. So corals are complicated. They're animals, but they are also in many ways rocks and plants as well.
Yeah, and maybe as a segue into a bit of the history of the Biosphere 2 ocean, I'd like to start by asking, this biosphere has a legacy. It's these systems that we're studying now are about 30 years old. And with that, this ocean has reached some sort of equilibrium state. And when did you guys decide to start to use this system for coral research specifically? And how is its legacy as a tropical coral reef in the past and now soon to be again? How does that kind of tie into this narrative?
Yeah, absolutely. I'll start that off and I'll actually pass it over to Katie, because a lot of that sort of seminal work that sort of brought us to where we are today started before I arrived on the scene. But I do want to show one thing that'll sort of set the stage for what Katie will talk about. And that's that, prior to this experiment, over sort of decades of collective coral reef research, we've sort of learned what we call sort of the building blocks of a resilient coral reef. And resilient coral reefs are reefs that can withstand changing ocean conditions and persist in the face of climate change. And so we'll get to coral reef health more in detail sure later, but we have learned what these key building blocks are, how algae play a role, how various reefs around the world fare. And Dr. Julia Cole and Dr. Ruth Gates, Katie, and a number of others around the coral reef community came together and realized that we have a really unique opportunity here at the Biosphere 2 to move from this decades of work on process, so what's happening and why it's happening, to really apply this and develop these solutions. So how can we build a resilient reef that will persist through changing conditions and exist for our children and our children's children? And so I want to pass it over to Katie to give a little bit more of that backstory. But really, I'm standing on the shoulder of those giants who had this vision for the project.
Yeah, so the Biosphere 2 ocean has an incredibly unique history. So the system behind me here is a 1 million gallon tank of salt water. And in the early 90s, you heard, if you were tuning into our last discussion with Jane and Tabor, they discussed actually going out to the Yucatan Peninsula to collect corals and fish and all kinds of animals to put into this reef system. So this ocean in the past has been a diverse tropical coral reef and survived at, inside not only the biosphere here, but at 4,000 feet elevation. So under a lot of different circumstances than they might see in the natural systems. And that reef was able to survive in here. So we know that's, data point number one for us is that we know that corals survive in this system behind us. And there's been incredible seminal studies that have come out of the Biosphere 2 ocean done by Columbia University in 1996. They really were looking at the effects of increased CO2 in our atmosphere and how that was affecting coral reefs around the world. So as CO2 goes up, we see our oceans really take up that CO2, and that negatively affects the calcification rate of these corals. So their ability to take ions that they build their skeleton with, they can't do it as efficiently with that increased carbon dioxide in the atmosphere. So we see them start to become stressed. And we were able to observe that right here in this system. We were able to see that stress response, which is called coral bleaching. You might have heard that term before. It's when the coral turns that stark white. So we saw our corals in the system under a lot of stress. Diane, so yeah, we've got a screen up here. So that middle section is that bleaching. So that's kind of what those corals look like when they're under that kind of stress. And after that experiment, we had seen, these corals be stressed, and over time and over different management, those corals went through different things, and we now today don't have any living corals inside of the Biosphere 2 ocean. So we've had over the years, over the last couple years, we've tried to decide what is the best way to use this incredible system that we have precise control over to be able to have an impact. And like Diane mentioned, Dr. Julie Cole and Dr. Ruth Gates were kind of the first people to come together and decide that this system can really be used to identify solutions that we can then put back into the natural systems to help save our coral reefs around the world.
Thank you, Katie. And some of our first questions coming through have to do with specifics of the ocean itself. Do you guys have a tidal system there? What kind of animals are in the water? So perhaps one of you or both of you could kind of give a general description of the Biosphere 2 ocean. How deep is it? What are some of the support systems that go along with it?
Yeah, I'd be happy to start that off, and I can pass it off to Katie as well along the way. So just to take a little bit of a step back here. So, we've both talked about how this system is so unique to test these solutions before they're applied in the wild. And that is because we have this sort of Goldilocks, if you will, of scale and control. And so fundamentally what makes the Biosphere 2 one of a kind for testing these solutions is that we both can test them at what we call ecosystem scale. And by that I mean we understand how these solutions play out from microbes, these tiny little organisms that we can't see, all the way up the trophic food web to the top sort of consumers. And everything in between. And so we really need to understand the system as a whole in order to understand which solutions will work in reality on the real world reefs. But the second piece of the problem is control. So we still need to be able to, at ecosystem scale, test very realistic conditions that we might see in the future. And it's often hard to pull apart these various drivers of stress that Katie alluded to, temperature and ocean acidification, so the decrease in pH of our world's oceans. How do we pull those apart and know what's driving, who did it, if you will? And so we have this Goldilocks of both scales, 700,000 gallons, but yet very precise control over the system. So coming to the question from Cara about tidal fluctuations, that's a great question, and one of the examples of control. So we don't have tides per se in the system, but what we do have on the far end of this system that you might be able to see here, and those of you who are in the previous webinar with Clay and Tabor, you saw, or Clay and Jane, sorry, you saw that they talked about the wave wall and the sound that it makes as they're on the beach. So this is a wave wall that produces realistic waves that would happen on a coral reef ecosystem. So that's one example of how we realistically represent a coral reef. Want to show you one other thing. So just to wrap that up, I want to sort of dive a little bit into the nitty-gritty, but I will walk through this figure. But one of the things that the creators of the Biosphere 2 did a really good job of, in addition to getting waves and realistic circulation on the reef, is they actually mimicked the bathymetry, the shape of the ocean, that looks a lot like a real coral reef. So those of you who have been to or seen images of the Great Barrier Reef and other barrier reef systems know that there are these sort of structures that line the world's coasts that look like what I showed earlier. So that's exactly what the shape of the Biosphere 2 ocean is. We have this shallow reef crest, a backloop reef lagoon, and a deep fore reef environment. And so this is actually really representative of what we see in the real world. So this back reef lagoon, it's a shallow reef crest, and then this deeper fore reef that goes to about 7 meters depth. So all that to say, we have this great scale, so 700,000 gallons. It's realistic, and we can very precisely control it.
And maybe pivoting on that to Katie, we've had a couple of questions as to what animals do you have in the system right now?
Yeah, it's a great question, and one we get a lot out here. When you think about the ocean, you automatically associate those animals with it. And we've been adding animals to our system recently. In the last year, we've really pushed to add organisms to the system. And the main reason right now that we've chosen certain animals is that they're herbivores, so that they eat algae. Our system, after those corals over time died off in the system behind me here, we got a lot of algae blooming, and that's pretty typical. It's what you see when coral reefs die off in the natural systems. You tend to see algae take over. So we have the same kind of reaction here. So we've been removing this algae pretty heavily in the last year. So we've removed about 10,000 pounds by hand over time, and that involves us going in and diving and putting them on these trays and sloshing it onto the beach and then taking it out of the biosphere. So it's a lot of manual labor. We get a good workout in those days. But we've also recruited some animals to help us keep that algae at a balance. And that's really important. We can't do all the hard work in here. We need something to kind of help us eat that algae as well. So I actually have some of my friends next to me here. So let me pull out one of my favorite animals that we've added. So this here is a short spine sea urchin. On top of him, he collects these little shells to help camouflage him here. But these guys we introduced last April, and they're amazing algae eaters. So they get in here and they help just kind of clean off those rocks. So these are one of the key animals that we've added. We've added them last April, and then we added more this past February. So when we go in here, we dive, we see them every time we go in, all on top of those rocks, especially on the reef area. They love it over there. One of the other a little bit smaller animals that we've added, but still equally as important, are these hermit crabs. I don't know if you can see him. He might be kind of hiding. He's camera shy. But these guys are also really great algae eaters. We've added thousands and thousands of these over the years, and they just really help us keep that algae at bay. Now, we also have about 7 species of fish in here, and some of those fish are descendants from the original mission or from Columbia's era. So last year in April, we actually added the first fish since 1996, and this was really, really exciting for us. We knew at this point our system was healthy enough to add animals like this back in. So you can see up on the screen at the bottom there, you can see the ocean surgeon fish. That is one of the animals. We added 22 of those guys. And then we had the red-banded parrotfish. Once we added 2 of those, and we plan to continue to add more species of fish as we move forward in this project to really help us keep that algae under control. A healthy balance of algae is good in a reef system. You just don't want it taking over the corals.
Thank you, guys. That gives us a really rich representation, both kind of structurally and from the perspective of what animals we have in the system. I want to move our gaze outward a little bit to kind of create a foundation for why we're doing research. We had one question from Kat about how long do we have before coral reef damage is considered beyond repair? And what are some of the more root causes of the need for research on corals in an experimental system?
Yeah, I mean, this is a great question. This is the core of why we're here and doing what we're doing. And so I want to give just a personal anecdote. I'm here because I personally have a tremendous amount of hope. So I'm newest to the project here. I've been here for about 2 years now. I came from Boston University, where I was before that. And when I heard about this opportunity to do coral reef research in the middle of the Sonoran Desert, I did not hesitate to apply because, like I said, we need to be thinking about solutions. But on a personal level, I've gotten here because I have not been without my share of seeing corals that I have grown to love completely decimated with local pollution and stress, as well as these major global drivers. So, these are some examples from some field sites that I work on. These particular reefs are in the Marshall Islands, which is in the Western tropical Pacific. A very small island nation that is going to be really heavily impacted by climate change and rising sea levels. So having seen reefs impacted by trash and covered in algae, following some of these really big stressors that Katie was talking about earlier, I'm very motivated to find solutions. But the other thing that I know is that we have seen evidence in the past that coral reefs can recover. The challenge is that we need to buy them time. So to sort of get to the answer to your question, one of the biggest challenges is that these bleaching events that Katie talked about are happening more and more frequently and more intense because of warming of our world's oceans. And when you combine it with the fact that as we release CO2, fossil fuels into the atmosphere, the oceans are actually taking up a large majority of that carbon dioxide. And with that, just like the CO2 that's pumped into our Coke bottles, our ocean is becoming more and more acidic. And this is really detrimental for coral reefs. So to answer your question, I've seen recovery on reefs. I have a tremendous amount of hope. We have colleagues all over the world who have come up with really unique solutions that I'm happy to talk about here in a little bit in more detail. And so we need to do 2 things. We need to buy corals time by addressing climate change, the elephant in the room. In the meantime, our team of 50 scientists from all over the world, we're going to come up with the best concoction of various solutions are to build the most resilient reefs we can all over the world.
And Katie, perhaps just briefly, you could chime in about your recent trip to the Great Barrier Reef in Australia.
Yeah, so I had the privilege to go to the Great Barrier Reef in February, and February in Australia is their kind of peak of their summer months. And we got to go out on the Great Barrier Reef. I hadn't ever gone before, so it was, a dream of mine to go out and see this reef. And it was as incredible as I thought it might be. It was so beautiful and so diverse, but as we are looking around at the corals in front of us, we also noticed what was underneath those alive corals was skeletons of corals that have been taken out in the very recent years, in the last 3 years, by these bleaching events. So for me, it was not only humbling, but also very motivating to see that, there is life on top of corals that have died because of these different stressors, but they were starting to get stressed out as well. So we, as a coral reef community, coming together and identifying solutions so that we can buy these corals time. If they are continued every year to be hit by these massive heat increases and they have to deal with that every year, we're just hoping to buy them some time so that we can continue to have reefs in the future.
Now, the current state of our Biosphere 2 ocean is a bit of a degraded one. And we say that because it had a fair amount of algae covering most of the surfaces. And while we've extracted a lot of that algae out by hand, and we have herbivorous fish and invertebrates working for us biologically, how is the degraded state of the Biosphere 2 ocean actually an advantage in starting to plan out research for testing resilient features of coral reefs?
That's a great question, and that's really the core of what we've been doing over the last couple of years. Is we're re-engineering and updating the life support systems. So for those of you who haven't seen the Biosphere 2 ocean in the last few years, just to put a bit of context behind what Katie was talking about, this is a 3D map, basically of stitched together photos that our collaborators at Scripps put together of what the Biosphere 2 ocean bottom looked like in 2018. So really every surface of the bottom of the ocean was covered in this algae that Katie described. So now this algae obviously has been removed. As she said, about 10,000 pounds of algae was removed by the team over the last year. But during that period, over the last couple of years, we used it as an opportunity to test what it takes to go from a degraded reef system like this one, which fortunately we haven't seen reefs that have been this degraded for over much of the world's reefs. But this could be the future of the reef if we continue to do nothing. So we had the opportunity to say, okay, what happens when we have a degraded system like this? How can we rebuild a healthy coral reef? So what we did is we spent the last couple of years studying everything that we could about this degraded reef system. Studying the physical environment, so what sort of environmental conditions, the temperature, the pH, the chemistry of the ocean, and of course, what the biology was doing. So everything from these tiny little microorganisms all the way up to the fish that Katie described. So we're really trying to understand natural cycling on this degraded reef and test what we call bioremediation solutions. So as it sounds, bio, organisms, so natural solutions for remediating or shifting that balance back towards a healthy reef. So we've learned a lot. In a couple examples shown here, we actually took a core. This is me and my PhD student, Alice Chapman. And this core of the sediments that accumulated in the Biosphere 2 since that original experiment that Katie described in the 90s, we have decades of sediment that's accumulated. And what we learned is we can look at the chemistry of that sediment and know why the system has shifted from this healthy ecosystem towards an algae-dominated system. And then, of course, an example here on the bottom of some of the biology. These are some of the key, what we call phytoplankton, so the plants that actually produce the base of the food web here in the Biosphere 2. And this is work that's being led by Dr. Jeremiah Hackett from University of Arizona.
Thank you. Diane, I'm really curious personally, and I think others are, about your background in geosciences and the way that you don't just study coral reefs, but you kind of deconstruct sediments of past oceans in some ways, right? Mm-hmm. Can you speak to how, to maybe some of that research that you do, but also how that plays into the biosphere as a kind of a proxy system for understanding these longer timelines of our ecosystems?
It's a fantastic question. And one I get asked a lot, and one that I also think about a lot myself, because just to take a really big step back, this is a compilation of pictures of me as a kid when I first saw a coral reef for the first time on the left on a family vacation to Buck Island National Monument in the Caribbean. To the right, where I'm now drilling holes in corals to study climate change. And if you would have asked myself as a kid, what I would be doing in the future, I can tell you it wouldn't be poking holes in corals to study climate change. So how did I get here, and how do we use this to develop solutions? Well, the short story is, on these trips, I was fortunate to see a healthy coral reef ecosystem, and then it was pristine, it was protected. But unfortunately, I returned in college quite a number of years later as an intern for the National Marine Fisheries Service, studying coral reefs, and jumped in the water, and my snorkel fell out of my mouth because the reef was completely gone. And so for the first time as a young adult, I saw firsthand what can happen to a coral reef because of climate change. So to answer your question, from there, my career took a hard right turn or left turn, whichever way, from coral ecology and biology to geosciences, because we can actually use corals to better understand these global scale drivers of stress on coral reefs. So now what we do, and this is an example from our work in the Marshall Islands, is we find these giant corals. Some of these can be the size of like a VW bus, either living, or in this case, a fossil colony, so one that was dead already when we cored it. And much like trees, they grow in these annual growth bands that you can see right here by eye. And so like tree ring research, we can use these rings of growth to look at what the ocean conditions were in that snapshot of time during which that skeleton was emplaced by that coral. So we get this rich history of ocean conditions over hundreds of years when individual corals lived. And so to bring it back to the biosphere too, then, what this work has shown us is that, first of all, coming back to my point about hope, is I've taken cores out of colonies like this and seen evidence that they died. Got covered with algae, covered with bivalves and other reef organisms. And then a couple years later, they pick back up and they start regrowing. And then sometime later, it happens again. So this evidence for regrowth and evidence of hope. But in terms of what we're doing at the biosphere too, one of the things that I learned from this work is that corals in regions that are hit really hard by stressors, so they are frequently hit by extreme events, they tend to do better than corals that are in very calm, stable environments. And so when I said earlier that we've learned about the key building blocks of coral resilience, that's this top building block. Is we know that corals here in this part of the Pacific in red that get hit really hard do better than the reefs further west. And that's a really key piece of information, because fast forward 10, 20 years, we now use this as a solution. So really smart colleagues have developed what we call stress hardening. So we harden corals, we turn up the temperature, we stress them out, and then we transplant them onto the reef. And they're more likely to be resistant to these stress events in the future. And we know this because of work where we core corals to look at past events. And so that's how it kind of all fits together and why we're doing what we're doing.
So is stress hardening something that you guys can do in the Biosphere 2 ocean? And what other forms of kind of resilient experimentation are you looking into to supplement stress hardening?
Great question. So absolutely. So that's going to be one of the main things that we test here, and among other solutions I'll talk about here in a second. And the way we'll do this is actually capitalize on what we call an experimental raceway system. So this is a 2,700 liter or 270 gallon tank, so much, much smaller than the 700,000 gallons in the Biosphere 2. And this is a separate system where you can see it has its own life support. So it has its own control to turn up the temperature. And we can use this system to harden the corals under different climate change scenarios and transplant them into the Biosphere 2 ocean behind Katie and say what happens to these corals at ecosystem scale. Which corals do get hardened, and how long does that resistance last? Does it only last 6 months, or does it last indefinitely? And we don't really know the answers to those questions yet, but we can test them right here at the Biosphere 2.
Thank you. One of our questions from Kendra was, what animals, if any, do you plan to add to the ocean biome? So maybe building off of stress hardening, starting with corals, but then going more broadly to other species of fish, other invertebrates. What is kind of the game plan for adding additional animals?
Yeah, so I will put this up as these are 4 of the species of corals that we have plans to bring to the Biosphere 2 in sort of the first set of experiments that we're doing in collaboration with the Hawaiian Institute of Marine Biology, so the institute that Ruth Gates was director of, led by Ty Roach in particular. And we are going to bring these corals and put them first in the raceway, experimental raceway, grow them, test various solutions, and transplant them into the Biosphere 2 ocean. Unfortunately, this was something that we were planning on doing in the coming weeks. And given the current situation, that's on hold a bit. So to be continued when that will happen. But nevertheless, we're cruising forward and are really excited to have a new postdoctoral fellow, Dr. Dan Killam, who is an expert in giant clams. And so Katie and Dan and I, we're going to, over the coming weeks, get giant clams into the system and start growing them to try to understand how they respond to stress in the Biosphere 2 ocean. Because much like corals, giant clams actually also have this small photosynthetic algae that live in their tissue. So we can understand a lot of the same processes by studying giant clams.
Thank you. We have a couple of questions, one from Andrew and another from Bill, and they kind of blend together well. Andrew asks, have there been any unexpected effects of increasing CO2 in the ocean biome? How was carbon introduced and measured inside B2? And Bill asks, have we learned much about coral bleaching from this lab? And I thought maybe this would be a good time to talk about the origins of the B2 ocean research, particularly the Columbia days, what that experiment was, but also talk about it in modern terms with how we would measure carbon dioxide in the ocean. And if we were to initiate any kind of other increased stress event, how that would be initiated into the system today?
That's a great question. So the good news is technology has advanced a lot since Chris Langdon's seminal experiments. So in these really seminal experiments that were done in the late 90s, he actually meticulously added acids and bases to reproduce the target pH, so the amount of CO2 that had been taken up by the ocean and ultimately changed the acidity of the ocean. And he did this to sort of mimic what at the time we thought was going to be the pH of the ocean at the end of the century, so at the year 2100. But we actually now know, if we continue, here is us in 2018. Unfortunately, as you can see, we are on the sort of worst case scenario emissions trajectory. So we are continuing as business as usual. And on this sort of red worst case scenario trajectory, we'll actually get to this condition that Chris Langdon reproduced in about the year 2050, so about 50 years sooner than he expected. And as Katie said, he showed definitively for the first time that corals were growing as much as 40% slower under these conditions. So this was really important work, and Chris Langdon is still involved in this project. And now in 2020s, we have the technology to actually bubble in CO2 gas and directly change the pH of the ocean. And this allows us to keep a very steady pH, whereas his experiment was kind of varying around some average value that he was targeting. So we're really excited to reproduce this experiment and more importantly, test these solutions and which ones work under these changing ocean conditions. And this is not related to your question, but kind of shows the advance of technology related to temperature control. This is what the latest and greatest, what we call a heat exchanger, that controls temperature of the entire Biosphere 2 ocean, so 700,000 gallons, at a rate of up to 2 degrees Celsius, or about 3.6 degrees Fahrenheit every single day. So we can really rapidly change the temperature of the system to really stress out the ecosystem if we want to reproduce these types of really extreme events that we expect to see in the future. So for reference, compared to this latest and greatest plate and frame heat exchanger that's made out of titanium, state of the art, there was an entire room in the back behind where you can't see behind Katie, where the old heat exchanger was. And so we've come a long way in the last couple decades, and we have a lot of technology on our side to really do groundbreaking research on coral reef resilience.
We have a couple of questions, one from Doctress and another from Leslie, that talk about some of the unexpected kind of positives of the coronavirus pandemic in terms of a decrease in carbon dioxide emissions, a decrease in pollutants. As a geoscientist, how do you think that coral reefs might benefit from this situational pause that we're taking?
That's a really good question. And to be honest, it honestly matters whether it is a temporary blip in behavioral change, or if we take this moment to pause and reflect and realize as a community that we have some profound work to do. Because as we've seen, for example, following the 2008 crisis, that there was certainly a blip in the emissions, and people started buying less fuel-intensive vehicles, and there was certainly a lot of positive changes that came out of it. But unfortunately, we tend to resort back to our habits very quickly. And if we do that, we will still be on track to continue to be on this sort of worst case scenario. So I think there's a tremendous amount of hope for what's happening in terms of the climate impact it could have. But that will only matter if we continue to have these major behavioral changes. And ultimately, a worldwide political sort of agreement to reduce emissions. So I love the optimism, and I really sincerely hope that that is one of the things that comes out of this really, really tragic situation.
And we have a couple of questions from Facebook, one from Garrett, one from Heather, that talk about some specifics in coral reef. One is, how long does coral take to die after it gets bleached? Another is the coral reef an endangered species? So maybe you guys could talk a little bit about the process of bleaching, how long it takes a coral to die, and then are corals recognized under endangered species acts in our kind of modern system?
Yeah, so I mean, the short answer to the first question is it really depends, which is kind of a crappy answer, I'm sorry, but it depends on how long, how intense the warming lasts. So this example that we showed earlier in the 2014 to 2015 extreme event that hit a lot of reefs really hard, a lot of reef loss happened within the course of a season. So you can see here from December of 2014, there's a healthy coral reef where you can see that coloration that comes with the photosynthetic algae. And then bleaching, so the skeleton is left behind in the clear coral tissue, so that's why it gets its name. And then it's a little tough to see, but by August of 2015, these corals are actually covered with algae. So even though they look kind of brown in color, this is turf algae like what we have in the biosphere too, that's growing over the coral. But in less extreme events, or if there is a gap in between individual extremes, corals can recover. They can actually, like if there's a part of the colony that dies, they can actually translate energy and nutrients horizontally because they're colonial organisms and regrow. So they don't always die, and it really just depends on how the event plays out. And then there was a second question, sorry.
Yeah. Endangered species, and are corals endangered?
So there are a few species of corals, particularly in the Caribbean, that have been put onto the endangered species list. Elkhorn coral being a classic example. This was I wish I had a picture in here, but it's very closely related. It's in the same genus as this picture here. But it looks like an elk's horn. So if you can imagine, it looks kind of like this. It's a beautiful species that used to be all over in the Caribbean and now has been almost, entirely lost across the Caribbean reefs. So there are examples of corals that have been put on the endangered species list. And more broadly, corals as a whole are really at risk, particularly on the trajectory we're currently on in terms of climate change.
And we have a question from Kat. She says she knows that acidification causes calcification among soft-bodied marine organisms. Does this have a major impact on coral reefs? And perhaps you could talk a bit about any kind of mutualisms that exist between soft-bodied organisms and the reefs that they kind of are a part of.
So I'm not sure I totally understand the question. Acidification causes calcification among soft-bodied organisms. So ocean acidification has a variety of impacts on corals of different types. It has the largest impact on hard corals. So the types of corals that I'm showing here that make this skeleton out of calcium carbonate, so that same mineral that that makes a shell on a beach. And that's because, like Katie said, that it impacts the availability of these key building blocks of their skeleton. Soft corals are certainly impacted in other ways. And to be honest, we are constantly learning about the complexities of various organisms' response to ocean acidification. And some have very complex relationships with this change. And so it's not always as straightforward of an impact as it is with these hard-bodied corals. But just to bring it back to why this is important, it's not that sort of soft-bodied corals aren't sort of as important for the reef, and obviously any impacts of ocean acidification on coral reef organisms is really critical. But the reason why we always focus so much on these hard-bodied corals is that they are really critical for these ecosystem services that reefs provide. So we often like to give this analogy between coral reefs, and this is something that Ruth Gates always did, and I love it, of comparing them to New York City. Like New York City, coral reefs are so biodiverse because of this complex 3D structure that these hard corals create, that produce these amazing nooks and crannies and habitats for reef dwellers. So just like our cities that have complex infrastructure that support these vibrant and growing populations, so do our coral reef organisms. And so really we focus a lot on understanding these hard-bodied corals because they are so critical for the biodiversity of the coral reef as a whole.
We have a question from Judy Andow. She's asking about the algae removal and if the U of A furlough and the coronavirus quarantine is an opportunity for people to get involved in continuing to remove algae, or perhaps more generally, we could talk a little bit about how people can get involved in the Biosphere 2 ocean research as a whole as well.
So in terms of diving, we definitely, people that are dive certified, we've had volunteers over the years that have joined our research team. We try to keep our groups going in the ocean at a minimum because the more people we have in there, the more we stir up the sediment. We try to really make sure we're not stirring and disturbing that sediment because we see an effect on our nutrients. So absolutely, if you're a certified diver, reach out to us. We have a list that we will occasionally reach out to if we have a big push. And in terms of right now, we don't have many people on site. We're practicing all the same protocols that everyone else is. Just a few of us are here making sure that the systems are still running. So right now, probably not the best time, but when we can get back to being together, that's definitely an option. And the best way to get involved is to reach out, if you're local, to reach out to us. We have volunteers that are part of the system that help us with daily tasks, but also just to follow along on social media platforms and our website. We have all these different ways to connect with us there and to follow along on the project.
And in the spirit of the current project, could both of you talk about what's to come in terms of adding infrastructure and continuing to develop the research here?
Yeah, absolutely. Obviously you saw that we've already come a long way in updating the infrastructure and have added a number of new organisms, as Katie described, to the system. So the next big step for us in terms of infrastructure is this other piece I didn't show here, which is this amazing truss of lights that can hold up to 288 lights to supplement the amount of light that reaches the Biosphere 2 ocean reef, because we know that it needs to be supplemented because of the space frame. And so that's going to be the next big project for us to tackle, and this will really ensure that we're able to build a really vibrant and healthy coral reef ecosystem in the Biosphere 2. But a little bit more broadly, we are really thinking about this project in terms of this sort of 3-phase structure. And we've talked a lot about phase 1 of this major project, which is where we've been in the past couple of years, which is really monitoring this degraded system to understand everything we can about how these degraded systems operate, how nutrients cycle, and things like that. Rejuvenate the system. So this is adding the key herbivores that Katie described, and then ultimately physically pulling the algae to clear the way for the coral phase of the experiment. And this is that bioremediation phase. So that's largely behind us now. We've finished phase 1. We've learned a ton, and we are excited working up some papers to publish on our results from that part of the experiment. And now we're working on this sort of engineering phase of building a resilient reef in the Biosphere 2 and starting to test these solutions for coral reef restoration. And so in the coming months, we'll start introducing corals. We talked about these giant clams that we'll bring in and start to build this diverse ecosystem. And then finally, in the 3rd phase, in the coming years, once the engineering is completely in place, we will sort of turn up the knobs, if you will. We'll try to mimic what we expect the future ocean conditions to look like on these reefs, and test these solutions under climate change scenarios, and really ensure that we come up with solutions that will ensure the persistence of reefs for generations to come.
And we are reaching the end. We've got a couple more questions. One was about artificial wave action, which we did talk about earlier. Wouldn't it be helpful in Biosphere 2's reef? Well, the answer is we do have some artificial wave action that's generated by a wave generator on the back wall behind Katie.
You can see it right now because I turned it off because it's pretty loud. So that's the reason the ocean looks so still.
Yeah.
It sounds like a dinosaur from Jurassic Park. And the other questions we have, which is an interesting one, is can coral reef be invasive? And perhaps you could talk about the idea of bringing in novel species into the Biosphere 2 ocean, and how that's both got its pro sides and con sides potentially.
Yeah, I mean, that's a great question. So to start with, the key piece of information here is that coral reefs are not all the same in terms of their response to climate change. So like many ecosystems, there are species that we consider weedy species. So even though they aren't invasive species per se, they can often dominate reefs following disturbance. And so you'll go to a reef where you'll see, actually I don't have a great example of that on this slide, but these sorts of branching coral species tend to grow really quickly and therefore often sort of take over, just like weeds in a forest, following these sorts of stress events. And so as Erin was alluding to, one of the big questions we want to look at is what sort of combination of species can create the most resilient reef that not only persists, but also still has these key ecosystem services. Because you can imagine, much like a field of weeds versus a forest, the services that those weeds provide is very different than that of the forest. And so we need this combination of diversity of species to give us that key societal benefit that reefs provide. And so the bottom line is we know reefs in the future will likely look different under stress, and so they might not be what they are today. But we need to balance having a diversity of species and being able to hit these key sort of functions that coral reefs provide, and make sure that we don't have just invasive types of weedy species take over the reef.
Diane and Katie, thank you so much for commemorating the 50th anniversary of Earth Day with us. And it's been a pleasure to have you on our last webinar of the day. The next event for Virtual Earth Day is a series of very special SARSEF presentations that you guys will all be able to find on our feed. And we appreciate all the listeners that we've had, and I can speak from my own experience that Katie and Diane and the team at Biosphere 2 in the Reef Lab are a group of very impassioned individuals that are really looking to solve problems in our oceans. So we're lucky to have a team like this with leaders like this. And thank you guys for tuning in today. We'll be back with more virtual events in the short future here. And you can follow all of our research on our social media, or look up more specifics at Diane Thompson's lab website. Any last words for our oceans and the Biosphere 2 oceans, Katie and Diane?
I would just add to, like Katie said, we are super excited to have people involved, especially from the local community. And even if you're not local and you're excited about what we're doing, please go to our website. We honestly couldn't do what we're doing without a tremendous group of collaborators and supporters. And so go to our website, and we would love to have your support if you guys are as excited about our solutions as we are. So we look forward to seeing you virtually or the next time at the Biosphere 2.
Thanks, everyone. Happy Earth Day.
Happy Earth Day. Take care.
Ever since I was a small kid, I wanted to learn how the world works. We're here in SARSEF. This is actually my first time here. I really wanted to do a science project. It's hard to even decide what you should even do your topic on. You can decide anything you want. My hypothesis is it will take the worms 16 days, and it will take the landfill 100 days. I actually used real worms. This is when I was actually at a real landfill.
This experience has been incredible. I think it's like a real game changer for our kids. He learned a lot about asking questions and making observations. I'd say about like 4 months in of like daily data recording, he got a little burnt out, but he powered through, and turning the project in was such a sense of accomplishment to him. When you do something hard and stick with it and you can be successful.
And you can just do anything.
I think it was a really good lesson for him. I've really seen him kind of apply the framework he's picked up through SARSEF to how he sees.
And I also had to go to a landfill.
And
but
yeah, you got
again, anything for science. ever since I was...
So the saguaro cactus is native only to the Sonoran Desert, and in the recent years, there's been this phenomenon where the central stems actually develop a bend. So this reference line here shows the tendency of the growth, and then you can see right here it starts to bend on this one as well, right about there. And so I was thinking that maybe heat is the contributing factor to why these saguaros bend, because the top is the new tissue, and if there's higher temperatures, it can weaken that tissue and cause the bend. So what I did was I picked 5 sites on the west side of Saguaro National Park and 5 sites on the east side. And I monitored the temperature for 25 days and then looked at the nearest 30 saguaros to the temperature monitor for each site. And so I used this pole here to calculate the degree of bend, and I used a compass to get the direction of bend for each of the bent saguaros that I found. And the data that I found with temperature was pretty striking, because an average of 5 to 6 of these saguaros per 30 were bent, which is about 1/5 of them, which is pretty significant. But the temperature doesn't really correlate with the amount of bent saguaros, because here we have, like, these warm ones are pretty big, yeah, but then this cold one is just as big as this hot one over here. This is the west side. And then on the east side, even more confusing, this big amount of bent saguaros is pretty cold, while this hot one is tiny. But what I did find is the tendency of the direction of the bent saguaros. So of all the bent saguaros, they were all bent south, and no outliers, which was pretty striking. And so from that led me to question, is it due to solar radiation exposure, since the sun goes across the southern side of the sky? And so a future study that I could do is to see if the south-facing hill versus the north-facing hill saguaros had different degrees of bend. Or I could just continue to monitor the bends, because I have the photographs and the coordinates of all of them, and basically just see if these bent saguaros are really surviving. So thank you for listening.
Alright, hi guys, my name is Sadie Northway Smith. I'm a senior at Tucson High Magnet School, and I'm going to be telling you all about my SARSEF project. So, we as a species have a trash problem. You guys all probably know this, but I'm going to talk today about a specific type of trash, styrofoam, which you guys all also probably know about, but it's really nasty stuff. It takes approximately 500 years to break down on its own, we think, and it's really, really horrible for the environment. We just keep making styrofoam, so we need a way to kind of deal with this buildup that we've created. And a couple years ago, scientists found a way to deal with that. In a pretty unlikely spot, they discovered that these little guys, mealworms, can actually eat styrofoam. These are the larval form of a darkling beetle, and the researchers discovered that their gut biome is actually what allows for them to break down styrofoam. So they have little bacteria in there that breaks down styrofoam entirely into biowaste. There's no plastic left over. But the researchers of that study, they didn't really investigate the real-world potential of their findings. You know, can we really use mealworms to break down styrofoam? So that is what I did in my study. I worked with 2 species, Tenebrio molitor, which is mealworms. Those are the worms that were used in the first study by the researchers, and then Xephobus morio, which are soup worms. That's this guy. That have not really been studied. So it was really exciting for me to get to work with those superworms. First, I established colonies of both species to kind of make sure they could both eat styrofoam, and I recorded a rate of consumption for both species, which was exciting. That was entirely new for Xephobus morio. I also had a pretty interesting finding. The styrofoam-only diet actually doesn't appear to inhibit the reproductive cycle of mealworms, so you can't really see in my colony here, but I have beetles in here, which are part of the life cycle of mealworms, and the worms in here are actually 3rd or 4th generation of styrofoam-only diet. So these guys have never eaten anything except for styrofoam in their life, and they're doing just fine, which is very exciting. But after I established those colonies, I isolated the gut bacteria from both species, and I grew it up in solution and added styrofoam. It was really cool. The bacteria was really, really beautiful. There was some that was this gorgeous lime green, and then another small percentage was actually neon orange, which is super cool to me. But from there, after I added the styrofoam and let the bacteria start to eat it, I kind of began to investigate what factors would affect that rate of consumption, things like temperature, types of incubator, concentration of LB broth that it was grown up in, to kind of try and find a solution to styrofoam waste on an industrial scale. I'm not quite there yet, just because the bacteria take so long to eat the styrofoam, but I'm working on it, and the bacteria do eat, which has some pretty cool potential in the field of bioremediation, which is very exciting to me. So thank you guys for listening, thank you to my teacher for his help and support, and thank you to everyone at SARSEF for fostering my love of science.
Arid environments may seem quiet and empty, but living in the desert, you likely know they are the home to an array of diverse plants and animals. Sadly, mining, deforestation, urbanization of natural lands, and fires are just a few of the human activities that are detrimental to the natural balance of these ecosystems. Experts work hard to deploy new methods and technologies to restore these environments. However, this is easier said than done. Replanting native seeds has many, many obstacles, some of which are dry environments where native seeds struggle to germinate, animals that eat and relocate native seeds, and invasive species that are highly competitive and steal nutrients and the little water native species have. Seed balls are a technology that show promise in the field of restoration. They are used to distribute seeds, but little research has been done on them. They are easy to make, being a mixture of soil, clay, seeds, and water. The mixture hardens, deterring seed-eating predators, and doesn't dissolve until the seed has sufficient water to grow. All of these attributes will help natives compete against the invasives. Our study looked at seed balls in a greenhouse. We planted different sizes of seed balls with different densities of seeds, and found the more seeds was better, regardless of the size of seed ball. Many studies about seed balls need to be conducted and replicated on different populations of native seeds to find the most efficient methods of restoration. These studies could look at how they're affected by different amounts of rainfall or how they're distributed in different ways. We'd like to thank Dr. Gornish for her guidance in everything from developing a question to analysis, Mr. Jonas for all his support, and Sarsef for giving us the opportunity to share our love of seeds, science, and the environment. Thank you for listening.
Hi, I'm Noah, and I'm a 5th grader at Twin Peaks KA, and I would like to tell you a story. When I was younger, I wanted to be a farmer. So my mom said I could make a farm in the corner of my yard. So I marked it off with big rocks, and I could see over it like I can with my engineering project. Let me tell you about it. It's called the TGOS, the Tethered Ground Observation System. It's basically a weather balloon and attached to a foam gondola. And let me tell you about it in 1 minute. First, let me tell you why I did this project. The reason I did this was because I was worried about deforestation. So I needed to make a project that poor land managers and people in poor countries could afford. I found out that they don't get paid very well. So I needed to make a affordable project because good photography drones are too expensive. And this project had helium in it, and that went up with the phone in this phone gondola that had an app that was set to take pictures for the time that you set it for. And for this, it's marked for every 50 feet that it goes up, and it went up like 250 feet, and the conclusion was that it was to help poor farmers and land managers help take pictures from their lands better, easier, and cheaper than they could with a photography drone. To make it as good as I want it, I could do some improvements. Some improvements might be to use strong fishing line because the paracord was too heavy, and I could let the foam gondola have more slack because it wobbled around too much, and I could put more helium in the balloon so it would go higher. And for a new engineering project, I could try to reuse the helium and the balloon because those were the expensivest parts of the project. Thank you, Sarsef and Biosphere, for letting me do this video. Bye!
Hello, My name is Kara Mayer, and my project focused on amyotrophic lateral sclerosis, abbreviated ALS, also known as Lou Gehrig's disease. ALS is a devastating adult-onset neurodegenerative disease. It is characterized by the death of motor neurons in our body, which control everything from blinking our eyes to breathing. Disease progression with such a loss in motor function is rapid, with most patients dying nearly 5 years after being diagnosed. Unfortunately, there is no known cure for ALS, and scientists are still unsure of exactly what causes it. However, in 97% of cases, they've identified a similarity. This similarity has to do with the protein in our body called TDP-43. In ALS patients, TDP-43 does not act properly. It goes out into the cytoplasm of their cells and accumulates. This accumulation of TDP-43 is related to the loss of locomotive function that people with ALS experience. Much of the information that scientists know about ALS and TDP-43 comes from studying Drosophila, or the common fruit fly. My science fair project investigated whether overexpressing UG-rich RNA, or ribonucleic acid, was sufficient to restore locomotive function in Drosophila with ALS. I looked at 2 different types of UG-rich RNA. The way I collected my data was through a method called larval turning. It can be a little difficult to explain, so it will work better if I just show you what it looks like. For the purposes of this demonstration, we're going to pretend that this ladybug eraser is a fly larva. What I would do is I would get the fly larva on a grape juice plate, and I would take a paintbrush, and using the paintbrush, I would turn the larva over, and then I would start a stopwatch. By its own means, the larva would turn back upright, and when it was back upright, I would stop the stopwatch and record the time. I turned healthy flies, I turned fly larvae with ALS, and finally, I turned fly larvae that had ALS in addition to UG-rich RNA. My results are shown here on this graph. As you can see, the healthy flies, the non-diseased flies, had an average turning time of a little over 10 seconds. The flies with only ALS had an average turning time of nearly a minute. As you can see, there is a huge difference between these 2 values, and it's clear that there was a loss in motor function. But when I turned the flies that had ALS in addition to the UG-rich RNA, we can see that the average turning time is closer to 20 seconds for the UG-rich RNA1 and about the same for the UG-rich RNA2. Since the turning time for the ones with ALS was nearly a minute and these were 20 seconds, there is clearly a statistically significant difference between these 2 values. Therefore, I concluded that overexpressing UG-rich RNA in flies with ALS did restore locomotive function. My project is extremely important, as before scientists can develop cures to any type of disease, they have to understand the disease on a cellular and a molecular level. My project is one step further in understanding ALS on the cellular level, which can hopefully and eventually lead to a cure for people stricken with this disease. Thank you.
Hi, my name is Sophie Brahim. So continuing on the work that I've been doing since the 7th grade, I investigated traditional Native American medicines on the bacteria that causes strep throat and kills nearly 500,000 people each year. Native Americans have a long and rich history of using plants as medicine, and so after interviewing tribal members and descendants from different southwestern Native American tribes, specifically the Pasco Yaqui, Salt River Pima, and Navajo, I was able to obtain the plant-based recipes for their medicines and test them in the lab. After making extracts of these plants, I found that 2 of the medicines killed one of which had no published information on its antibacterial properties. This was a novel discovery and very exciting. In the future, I hope to continue this and work and help develop new antibiotics that could potentially help the antibiotic resistance crisis. Thank you.
Hi, my name is Layla. I'm a 5th grader who loves hiking, riding my bike, and camping with my family. Every year we go camping up on Mount Graham. My family has been going to this mountain for over 70 years, so a long time before I was born. A few years ago, we had to cancel our family camping trip for the first time ever due to the wildfire on Mount Graham. The year after the fire, I couldn't believe what I was seeing. When we drove up the mountain to our usual camping spot, my heart was broken as I saw so much of our beautiful mountain burned and lost. Wildfires are a problem everywhere in Arizona and many other states as well. When trying to come up with my science project for this year, I wanted to find a way to stop the spread of wildfires in Arizona without having a negative impact on the environment. I chose this project because I am passionate about finding creative ways to control wildfires that everyone can agree on. The goal of my project is to figure out which type of native Arizona tree burns the slowest. In an attempt to control wildfires and decrease the chances of future wildfires in Arizona, especially in burned out areas, if we can plant more slow-burning trees that are native to Arizona so they grow in the wild with little to no maintenance, the slow-burning trees will slow down the rapid spread of the fire compared to the trees that burn quickly, allowing firefighters a greater chance at controlling and putting out the fire. When considering what trees to test for my project, it was important for me to only use trees that are native to Arizona because they would survive better in a natural environment on their own. During my research, I found that mesquite, palo verde, and ironwood and acacia trees use the least amount of water among the native Arizona trees I studied. I ended up choosing 3 types of native Arizona trees: ironwood, palo verde, and mesquite. To test how slow samples of wood from each type of wood would burn, when it came time to do my experiment to control the volume of the wood that was being tested, I chipped up wood samples from each tree for each trial. I placed 1 wood sample in a fire pit, lit it, and timed how long it took for the sample to burn out. From my data, I found that ironwood burns the slowest and palo verde wood burns the fastest. It is interesting to note that although ironwood took the longest to burn, it also lit the fastest. In contrast, the mesquite wood took the longest to light, but did not burn the slowest. This raises an interesting question. When trying to prevent wildfires, is it better to have slower burning trees, or would that take longer time to light? The purpose of my experiment was to find which wood would be the best to use to replant a burned out area after a wildfire has come through. To decrease the chance of future wildfires, at the beginning of my experiment, I was looking for the slowest burning wood. As a way to decrease the chance of future wildfires, using this theory and the result of my experiment, the ironwood tree would be the best tree to use for reforestation because when it catches on fire, it burns slower. The wildfire is most likely not to spread as quickly because the ironwood tree burns the slowest, giving firefighters a great chance of putting out the fire and minimizing the damage. However, this experiment inspired me to further investigate the purpose of my project, which is to find creative ways to control wildfires and everyone can agree on. I would like to do further experimentation and research to determine if slow burning trees or slow lighting trees would be a better natural defense against the spread of wildfires. Since these turned out to be 2 different types of trees, my project is important because of the potential positive impact I can have on the environment. Reduce the spread of wildfires by planting slow burning trees instead of deforestation. Reducing atmospheric carbon dioxide, improving air quality, protecting water and soil resources, and reducing runoff. Thank you for listening.
Hi, I'm Noemi, and I'm really interested in conservation for endangered animals and plants. The vaquita marina is a small porpoise that lives in the Sea of Cortez. It is the world's most endangered marine mammal animal, and scientists think there are less than 15 left. One problem of advocating for the vaquita is there are not really very many good pictures. There are very few of them, and they avoid people in boats. So how do you get people to care about a plant or an animal they can't see? Most importantly, how do you get them to act? Research shows that people learn better when they have a picture and text. They also react to brightly colored images. For my project, I gave 3 groups of students information about the vaquita. The first group had just text. The second group had text and a picture of a vaquita's dorsal fin. And the third group had a lifelike painting of a vaquita. Students answered questions including how much free time would they be willing to give up to help save the vaquita. The lifelike painting made an impact. Students who got the text along with the lifelike painting said they'd give up more free time to help save the vaquita. This information will help me and others who are working on plant and animal conservation. Now we know how to catch people's eye and interest. Even more, it helps us know how to inspire others to take action. Seeing means acting. In the future, I hope to do more research on this topic, like what happens if the plant or an animal is not cute, and do adults respond the same to images as kids, and would kids be more interested if there was an activity along with it, like a coloring page. I'd like to thank Viva Vaquita for their dorsal fin photo and Dr. Julius Chutney for his painting. Also, I'd like to thank SARSEF for their help and support and providing me a way to share my science.
Hi, my name is Mason Albright, and this is my 5th grade science project. I chose to put it in the environmental engineering category. My project is about finding an alternate use for plastic bags so they don't end up in landfills and oceans. I came up with this project when I came home from school one day and I was watching a nature film about plastic bag pollution, and I knew I wanted to do something about that and fix it. Did you know that 1 trillion plastic bags are thrown away every year? That's 2 million each minute, which means most of those are going inside sea turtles and dolphins and making them very sick.
And I made a little pulley system, so when you spin one hook, the rest of them spin.
So I don't have to do the 6-person twisty thing. I put a screwdriver here and twisted it. And this
year particularly important because it's 25 years since the scientists went to live in the biosphere for 2 whole years, can you imagine? And 2 of them actually got married. And these 2 went on to form one of the US's most innovative companies, Worldview, which is radically thinking the way we act.
We need 3. Now, this should fit on there. Okay, we need to cut this 7.5 inches. So, right there. And I need to line this up with the other one. There. Okay, put this together. I'm gonna show you how I did it. I put a cut washer, then a nut, then another cut washer, and then a medium-sized washer, like this, and then one of these, then a wheel. These are my ropes. There's this rope, which is one of these little things.
braided to make this rope. And I don't believe it worked because when it was braided and it was being pulled, it had a tension point when it was being braided at places like here and here. And this is by far my worst rope, little crocheted pieces, which created so many tension points that it basically just snapped automatically. This is my best rope because it was making the weight distributed in between all the different rope pieces. And here's a closer shot of the rope. This is by far my favorite one, and it took 418.8 pounds to break it. And this is my control rope made out of natural materials. They killed plants to make this rope. And my red rope did way better than that rope. Why are we throwing away material that could be used? My project proves that plastic bag rope is just as good or better than regular rope. People should start treating plastic bags like material instead of waste. While we're all stuck at home, maybe we can think of ways to better the environment. Maybe after this virus, we can come back to a better Earth. This is Mason Albright. Happy Earth Day.
So the saguaro cactus is native.
Arguably, some of the most significant research to come out of Biosphere 2 originally came out of the ocean. So this system was one of the first places to show a direct connection between increasing atmospheric CO2 and the negative impact that it has on coral calcification rates.
We knew that our coral health around the world was declining at a rapid rate, and we were able to connect with Ruth Gates in the Gates Lab at the Hawaiian Institute of Marine Biology, and she was doing incredible, innovative work looking at how to identify corals that are more resilient to these temperature changes that we're seeing happening. And she was doing it at a lab scale, and she saw our system, this million-gallon tank behind me here, and her entire body lit up saying, we could do science here that we can't do anywhere else in the world. And from that point forward, our focus was on how to renovate this system and bring it back to life so that we can do life-changing studies looking at coral reef ecology.
The first thing we needed to do was we needed to modernize the heat exchanger, that unit that allows us to heat and cool this water. Now what the researchers can do is they can call up and say, I want to increase the ocean temperature by, for example, 2 degrees C, and I want to do it in a 24-hour period. We can do that super precisely.
When reefs die off around the world, algae tends to take over. Those species that are around coral reefs that tend to keep algae away usually also go away when coral reefs die. So you see these mass algae blooms occur, and pretty much every surface is coated with algae. And that's what we had out here at the Biosphere 2 Ocean for some time. And in the last 6 months, we've started to extract that algae, and so we've got teams of divers out here, our research staff that goes in and actually hand pulls this algae out. We've pulled about 10,000 pounds of algae out by hand in the last 6 months.
The second thing that we're looking to do now as part of this engineering and design was to look at, okay, if we want to grow corals in this system, one of the things that we need to do is we need to make sure that we've got enough light for them to grow. The latitude that Biosphere 2 is at is a hindrance in some respects for growing corals if you're just using natural light, because it's much less than that you would find in a lot of these Caribbean or our tropical regions. So what we can do is we can actually supplementally light the system. We can add trusses over it and very strategically place these additional lights. So that's going to be phase 2. Phase 1 is this heat exchanger. Phase 2 is going to be adding the necessary infrastructure so that we can light the system. Now what's really exciting in conjunction with this though is the team has been very closely looking at the water chemistry. And again, they've been able to confirm that we have a very stable system, and that now what we can do is we can look to make new introductions into the system to build the complexities of the community that we have to complement the type of research that they want to do.
Last year, April 2019, we introduced 22 fish. This was the first fish introduction since 1996. So it hadn't been up until this point that we felt the ocean was healthy enough to reintroduce a species like that. So we introduced 2 species. We introduced red band parrotfish and ocean surgeonfish. And these fish have been in here living in the system for a year now and have been really successful. So we have about 7 species of fish, over 40 individual fish in here right now, and we will continue to add to that as time goes on to develop this community that we want. Other than fish, we have some invertebrates, and these invertebrates are incredibly important to the system. We've got some red-legged hermit crabs, top-crowned snails, short-spine urchins, and all of these animals were introduced to really help us eat the algae in the system. We have our first coral raceway here at the Biosphere 2, and this raceway is a 700-gallon tank that has a full life support system, and it's completely isolated from the Biosphere 2 ocean behind me here. So it allows us to bring in corals, put them into this tank, and really try these experiments out, and then be able to put them into the ocean here. So one of those experiments that we've been discussing with our partners is stress hardening. So we put these corals under stress over and over and over again, and they get stronger every time. So you push them just to their limit, and then you back it off. And then you push them a little bit further next time, and then you back it off the heat. So it allows us to strengthen these corals.
Right here in the Sonoran Desert, we have a team of scientists that are going to give significant insight to helping this global challenge and this crisis that we see with corals around the world.
Particularly important because it's 25 years since the scientists went to live in the biosphere for 2 whole years, can you imagine? And 2 of them actually got married. And these 2 went on to form one of the US's most innovative companies worldwide.
All right, Aaron, you're live.
Radically thinking the way we act.
Hey guys, my name is Aaron Bouget, and right now I'm filling up what we call an Oya bottle. The concept of this bottle is about 4,000 years old. People have been using terracotta clay as an irrigation source for their gardens for thousands. And as I fill this Oya bottle up, you'll start to slowly see some black spots
This year, particularly important because it's 25 years since the scientists went to live in the biosphere for 2 whole years, can you imagine? And 2 of them actually got married. And these 2 went on to form one of the US's most innovative companies, Worldview, which is radically thinking the way we access space. So would you please welcome Jane Pointer and Tabor McCallum.
The fundamental unit of life is a biosphere. All life as we know it is within the context of the biosphere, and we have but one of them. So Biosphere 2 was meant to test the idea that we could create an artificial biosphere, a small biosphere, out of the ingredients of the only biosphere that we knew.
25 years ago, Tabor and I went into the biosphere to begin the first 2-year mission. I am doing the Queen's Wave there. And Tabor, unfortunately, has his arm over his face. But anyway, there we are. We were going in to test this biosphere to really see, does it work? Is it possible to take a biosphere on this incredible planetary scale and have it survive for an extended period of time? So you've seen all of the biosphere, so I'm not going to talk about any of what is in there except the part that you haven't seen, really, which is the agricultural system. Here's the farm. I was actually in charge of the farm inside Biosphere 2, so I was in charge of growing all the food. So when you think about Biosphere 2, we had to take care of an entire world, right? So one of the things we had to do was grow all our food. We also had to recycle our water. We also had to manage all our atmosphere. You know, in many ways, it's just really what we're having to do on planet Earth today. So here's our farm. You know, we knew at every moment where all the food we were eating from came from. We knew exactly what was in our food. You know, we were incredibly connected to our food, to the animals. You know, it took us 4 months to make a pizza.
By living as part of our little biosphere and seeing another biosphere outside Earth's biosphere and having this sort of intra-biospheric communication, we would say, you know, what is our CO2 today? And they'd outside in the Earth's biosphere, they'd say what their CO2 was today and what our oxygen was and what their oxygen was, and have 2 biospheres that we could sort of compare and understand. You know, we would get weather reports in the morning and in the evening, what the CO2 was and the oxygen was and temperatures. So we were really in touch with our little biosphere, and it's a very similar, it seems, experience to what astronauts talk about. So we've taken this idea to a company called Worldview that Jen and I and others started, and the idea is to do 2 things: to understand our planet using high-altitude balloons. These are very extremely low environmental impact way of getting instruments into very critical parts of the Earth's atmosphere for long durations to understand global change and global warming and the chemical balances that are occurring in our atmosphere using high-altitude balloons that fly for long periods of time, but also using this same technology set to take people to 100,000 feet. And the reason we think that that's really important is a lot of what we've been seeing today is getting that visceral understanding that we live in a relatively small ball. So this is a flight that we did last year at a scale flight of our spacecraft going up to 100,000 feet. And what's important to think about this is that we're crafting a business that every aspect of it, we think about what's the environmental impact of that? What's the environmental impact if we scale the technology that we're developing to a large scale? And I think the thing that we all need to think about, that we've heard a lot about today, is I don't think businesses are going to succeed going forward unless they're thinking about the overall environmental impact that they're having, the way we train our people, the way we educate our people. And that's really going to be the key, I think, to really seeing the future and building the kinds of companies and organizations and institutions that will do well going forward. This is looking at from about 100,000 feet. The lens made the Earth a bit backwards on this one, but it's a great image. And then after we've taken the samples we need to take, we've done the research we need to do, we fly back down under parafoil to get the payload back down to the location that we want it to be. And the whole system is essentially solar powered. While it's flying, we put out solar arrays, and the system is driven from the power it's getting from the edge of space.
This year, particularly important because it's 25 years